Okay, I'll share my screen with you and we'll start today's class.
Sorry, ma. Okay.
Let me know if
I can see the screen. Basic concepts of microbiology.
can you see my screen?
I can see the screen.
Yes or no? Can you see my screen?
Yes, I can see your screen.
Okay, fine. So today we're going to learn about the basic concepts of microbiology and parasitology. Okay,
Okay.
this is what we're going to microbiology. Microbiology. What is microbiology? That learn. So if there is any doubt, then you can and there is any doubt you can ask me the doubt. Understand. Okay. Can you? Yeah. Thank you. So if there is any doubt, you can unmute and you can let me know. Okay.
Okay, mom.
So today's class, we're going to learn about the microbiology. So first we'll understand what is microbiology and what is its role in the day-to-day health and the disease. Science and putting the So what do you mean by microbiology is a study of different kinds of microorganisms. And these microorganisms will include different kinds of bacteria, viruses, fungi, and proteins. Okay. Organisms everywhere ecosystem. And they play a role in the day ecology, the disease. That is why there are different microorganisms.
The network of today is really bad. Really bad.
Sorry.
I mean, the network, it's really bad. The voice is coming in segments.
Now can you hear me?
Yes, I'm hearing you.
So we are discussing about what is microbiology. Okay.
Okay.
It's nothing but study of different kinds of microorganisms. Okay. And these microorganisms will include different types of bacteria, different types of virus, different types of fungi, and different types of protozoa. We'll discuss each one in detail. Now, these microorganisms do exist in the everywhere and it plays various essential roles in our healthcare system, ecology, as well as in the disease process. So, why it is important to understand microbiology is to provide a better patient care, to prevent the infections, and to promote the public health. That is why, being a nurse, you need to learn about the microbiology. Is it clear?
Okay.
Is it clear?
Very clear, mom. I'm getting you.
So these are the different microbes we will be learning in detail. As I told you, there are viruses, bacteria, fungi, and parasites. These each one we will be learning in detail. Okay?
Okay.
So very start with cell. As you have learned in the biochemistry also, everything begins with a cell, right?
Yeah.
What is a cell? It is a basic structural and a functional unit of a life. So everything and
Of
anything
life.
is made up of the cells. Okay? Cells are the
Okay. Okay.
structural and the functional unit of a life. And this to compare the microbial cells also with the human cells. So now we will learn what are microbial cells and how it differ from the human cells. Okay, fine.
Okay.
Uh, We'll start with the microbial. So single cells synthesis the life. Understood? So microbial cells are the microscopic, a single celled organism, and they represent the simplest form of your life. And they exist independently or in colonies and are responsible for a wide range of biological processes in nature. So they do live in the nature either independently or by forming colonies in a group. That's how they live in the nature, and they are biological process. Understood what is microbial cell? So microbial cells are the microscopic single-celled organism which represents the simplest form of the life. And they exist either independently or in a group, colonies. And they are responsible for various biological processes. Biological process in the sense decomposition, fermentation, like that various processes they are responsible. Is it clear what is microbial cell? Is it clear? Is it clear? Is it clear?
Yes, it's clear now. It's clear.
Now we'll see the structure of a microbial cell. Fine. We'll see what is the exact structure of a microbial cell. We can classify microbial cell into two types. One is prokaryotic and another one is karyotic. So it can be divided into one is prokaryotic and eukaryotic and the prokaryotic cells. Now, what do you mean by prokaryotic cell? These are the ones which do not have a nucleus. They lack the nucleus. So if you see a prokaryotic cell, you can't see any nucleus. So that is why they are classified as prokaryotic, where they don't have any sort of nucleus. They lack the nucleus. And DNA is present in the nucleoid. So in a cell, there are different organelles present. And when you classify a microbial cell, you can classify into prokaryotic and eukaryotic. And what you mean by prokaryotic is which they do not have a nucleus and their DNA will be present inside the nucleoid. Understood? Then
Yes, ma'am. Yes.
contains a prokaryotic cell will contain ribosome, a cell wall, and a plasma membrane also. Okay, so usually a prokaryotic cells will not have any nucleus. Their DNA will be present inside the nucleoid, and the prokaryotic will contain ribosome, a cell wall, and a plasma membrane. And they have spindly flagella or a cap, like tail kind structure is what we call it as flagella. Understood what is prokaryotic cell?
Yeah, yeah.
Prokaryotic cell is bacteria or archaea. These are the different types of prokaryotic cells. And this is what prokaryotic cell will look like. Is it clear?
It's clear. It's clear.
Yeah. Next we'll see what is eukaryotic cell. So see, this is how an eukaryotic cell will look like. It has got different things inside the cell, right? So eukaryotic cell example you can see is in the fungi as well as in the protists and all you can see eukaryotic cell. Now what is the difference you can see in the eukaryotic cell is they have a nucleus and a membrane bounded organelles. See as you see here, it has a center nucleus and different other organelles are present inside. So that is how eukaryotic and prokaryotic will be different. Understood? Now, eukaryotic cells has different organelles like ribosomes, mitochondria, endoplasmic reticulum, and Golgi apparatus. And they also have a cell wall or a motile structure like cilia here. So, eukaryotic cells define this in the center, which is not there present in the prokaryotic. So, eukaryotic cells are the one which have a defined nucleus and other membrane organelles like ribosomes, mitochondria, endoplasmic reticulum, Golgi apparatus. Everything is present in a eukaryotic cell. And they also have a cell wall. Outer layer also they have cell wall and other motile structure like flagella or a cilia.
So the network of today is really, really poor.
Sorry?
I mean, there's bad connection today.
I can hear you. Sorry.
I said the network here is really, really bad today. I guess that's why others are not here.
But can you hear me now?
The network is bad.
Can you hear me now?
Yes, I'm hearing you. Just be teaching, Mom.
Okay. Fine. Can you see my slides? Can you see the slides?
Yeah, I can see the screen. I can hear you.
Okay. So cooperate with me and just you also just read the slide. If there is any doubt, you can ask me. I'll repeat. Okay?
Okay, Mom. Okay, thank you.
Yeah. We are discussing about the eukaryotic cell. You understood what is eukaryotic cell? Eukaryotic cells are the cell which has got a defined nucleus and the other organelles. The other organelles include cytosol, endoplasmic reticulum, Golgi apparatus. Okay?
Okay.
Now we'll see what are the functional aspects of a microbial cell. That is what microbial cell. The microbial cell, either a binary fission or a budding. Binary fission means you can see in the amoeba, a single cell will divide into two different. Or budding means from a parent plant only there is a bud formation and reproduction happens. Like example in the hydra, budding in the hydra and binary fission in the amoeba. Understood how they perform the biological function? Reproduction is through binary fission of budding cells. Is that
Okay.
yeah? Second, biological function they perform metabolism. Metabolism, like the breakdown will occur in the presence of oxygen. It is aerobic, absence of oxygen, anaerobic, and fermentation. So that is how bacterial metabolism takes place. Anaerobic in the presence of oxygen, anaerobic and fermentation in the absence of oxygen. Clear metabolism?
Yes, Mom. Yes.
Yeah. Third one is exchange. Like the genetic factors, RNA, DNA ka exchange will occur either by conjugation. Conjugation means in the figure you can see they form a connection and through that connection the genetic materials will be passed on from one body to the other. That is what conjugation. Second one is transformation. Transformation means directly from one body to the other they will transfer. And transduction. Transduction from That is, from the one body, they will pass on to the other. The genetic materials will be passed on from the one to the other. So these are the different methods by which there will be exchange of genetic materials like DNA or RNA. Then their role in the ecosystem. As I told you, microorganisms are required in the decomposition of waste, nutrient cycle, and in symbiosis. So these are the different functions a microbial cell will be performing. One is reproduction, one is metabolism, one is genetic exchange, and the last one in the ecosystem also it helps. Understood the functional aspects of a microbial cell? Is it clear? Functional aspects? Functional aspects is clear?
Yes, it's clear.
So this is how a prokaryotic and a eukaryotic cell will look like. So prokaryotic cells are the one which do not have a nucleus and the organelles inside. Whereas eukaryotic are the one which has a defined nucleus and all the organelles are present inside the cell. Understood? And prokaryotics are simple and eukaryotic is a little bit complex. Understood how prokaryotic and eukaryotic cell will differ? Just see the picture. Just see what all things are there inside eukaryotic and prokaryotic. Just see once.
Yeah, see the picture.
Yeah. Is it clear?
Yeah, it's clear.
So in this chart, I have given what are the difference between a eukaryotic and a prokaryotic cell. Okay. So we'll see one by one. Can you see this? So what is eukaryotic? Any?
Yes, I can see.
Can you see?
I can see definition.
Now can you see?
Yes, I can see.
Yeah. So the very first one is the definition of a eukaryotic cell. So eukaryotic cell is what? The cell which contain a clearly defined nucleus and membrane bound organelles. If you see a cell with a nucleus and different organelles, you have to see that it is a eukaryotic cell. Understood? Whereas
Understood. I'm following, Mom.
here,
Clear.
yeah. Whereas when you see there is a unicellular organism which do not have any organelles and a nucleus, then you have to characterize them as prokaryotic cell. Okay. Now,
Okay.
examples of eukaryotic cell is animal cell, plant cell,
Plant
fungi,
cells.
whereas prokaryotic can give example and now nucleus is present in the eukaryotic cell and nucleus is absent in the prokaryotic cell. Cell size, if you see, eukaryotic cells are larger in size and prokaryotic cells will be smaller in the size. Now, if you see DNA replication, it is highly happening in the eukaryotic and replication will be only at once occurring in the prokaryotic. Now, if you see organism type, usually your eukaryotic will be multicellular and prokaryotic will be a unicellular. Prokaryotic will be unicellular, eukaryotic will be a multicellular. Chromosomes, more than one chromosome you can see in a eukaryotic cell and only one single chromosome or a DNA will be present in the prokaryotic cell. If you see a ribosome, large size of ribosome will be present in a eukaryotic and a small ribosome will be present in the prokaryotic. Now growth time, if you see, eukaryotic cells will be slower in rate, whereas prokaryotic will be faster. Now presence of organelles, as I told you, in eukaryotic you can see organelles are present, whereas in prokaryotic organelles will be absent. Now, ability to store their hereditary information. All the eukaryotics has that ability to store the information. And prokaryotic also have that same ability. Now, if you see the cell wall will be a simple in presence of eukaryotic and cell wall will be a little bit complex in case of prokaryotic. Then plasma membrane is present in the eukaryotic and it is present in the prokaryotic. Cytoplasm is also present in eukaryotic and prokaryotic. So these are the major difference. If a question comes like explain the difference between a prokaryotic and eukaryotic cells has been asked, you can mention this. Clear? Is this clear? Is this clear?
Yes, ma'am. Yes, it's clear.
Okay, fine. Now we'll discuss which are the different types of microbial cells. Okay, one is bacterial cell. As I told you, they are prokaryotic. They do not have any nucleus. And they are diverse in the shape. There are different types of bacterial species are there in different shapes, either in a cocci, bacilli, and spirilla. These are the different shapes available in the nature. Bacteria will be prokaryotic cell and they will diverse in the shape, cocci, bacilli, or spiral. These are the different types. Second type of microbial cell is archaea. Archaea is also again a prokaryotic cell which do not have a nucleus and they are extremophils with the unique biochemistry. Extremophils means they live in a extreme conditions. These kind of microbes will live in a extreme conditions. Third one is fungi. Fungi are eukaryotic. Eukaryotic means they have a defined nucleus is present and they can be either unicellular or they can be a multicellular. Next type of microbial cell is protist. They are also eukaryotic with a nucleus present and they are also diverse group include algae, protozoa. There are various varieties are there. The last one is virus which are acellular. Either they do not have a true cell, but they can infect and replicate in a host body. Viruses usually enter the body and they will multiply in the human body. So that is what they can infect and replicate in the host body. So these are the various types of microbial cells which we're going to discuss in detail. Understood? Types of microbial cells, bacteria, archaea, fungi, protist, and the virus. Types of Understood? Understood different types of microbial cells? Understood what are the different types of microbial cells? Understood? Is it clear? You got to know what are the different types of microbial cells? Understood?
Yes, understood.
What is a human cell? Till now we discussed a bacterial cell. Now we'll see what is a human cell. Okay. So human cells are also eukaryotic. As you see in the image, there is a nucleus present and there are different organelles are also present. So human cells are eukaryotic containing a nucleus and membrane bound organelles. As you see in the image, there is a nucleus and different organelles are also present. They are specialized for various functions. In the body, they conduct nerve conduction, muscle contraction, and immune response. So in a human body, this human cell has got different function. They help in the nerve conduction, muscle contraction, as well as the helps in immune response. And human cell is a part of multicellular organism and communicate through signaling pathway. So different signalings will happen in the body and that's how human cell will be communicating. Understood what is human cell? They are eukaryotic cell with a nucleus and different organelles and they have various functions like nerve conduction, muscle contraction and immune response. So this is what a human cell will look like. Understood what is human cell? Understood human?
Yeah, understood. Yes, I'm getting you.
So see what is the structure of a human cell. So key features if you see in a human cell, one is it has a nucleus which will be containing the DNA and the genetic materials. Second thing, a human cell will have a plasma membrane with a phospholipid bilayer. It is the outer covering the human cell has got. It is a plasma membrane with a phospholipid bilayer. Then it also has got organelles. Organelles like mitochondria for energy production, endoplasmic reticulum for any protein or a lipid synthesis, and Golgi apparatus for the protein modification. And they also has got specialized structures like cilia and cytoskeleton for their support. Cilia helps in the movement and cytoskeleton helps them to support the structure. So these are the different structures you can see in a human cell. One human cell has a nucleus which contains DNA. Second thing, it has an outer coating like plasma membrane, which is a phosphobilipid layer. It also contains human cell contain organelles like mitochondria for the energy production, endoplasmic reticulum for the protein or lipid synthesis, and Golgi apparatus for the protein modification. And it also, human cell also contains specialized structures like cilia for the movement and cytoskeleton for the support. Okay.
Okay, ma'am. Okay.
Yeah. Now we'll see what is the major difference between a microbial cell and a human cell. So difference between microbial and a human cell, first thing is in the size. Usually your microbial cells will be smaller when it is compared to the human cells. Second thing is nucleus. Like microbial cells are usually prokaryotic. That means they do not have nucleus, lack true nucleus, whereas your human cells will have a defined nucleus. Third one, reproduction. Microbials will reproduce quickly, either through binary fission in the bacteria, whereas human cells will divide slowly. Next one is cell wall presence. A cell wall is present in the bacteria and in the human cell it is absent. Then presence of genetic material. Human cells have a organized chromosomes. Microbes will have plasmid. So these are the major difference you can see in the microbial and human cell. So again, microbial cell will be always smaller when you compare the human cell. Then human cell have a defined nucleus, whereas the microbial cell will not have any nucleus. Reproduction will be happening very fastly in the microbial cell and reproduction will be slow in the human cell. Then cell wall is present in the microbial cell and it is absent in the human cell. Genetic materials are present in the human cell in a proper organized chromosome, whereas microbial cell, they have plasmid in which the genetic material will be present. Is it clear?
Yes, ma'am.
Now, what are the functional comparison? If you see microbial cell, there is a rapid growth and reproduction, whereas in the human cell, there will be slow reproduction, you can see. Then diverse metabolic pathways are available in the microbial cell. For example, anaerobic respiration, aerobic respiration, and fermentation. Like in presence of oxygen, in the absence of oxygen, like different pathways are available for the microbial cell. And microbes can either be a free living or they can live in a colonies or by living in some other, they can perform or they can live. Whereas human cells, they have specialized function. As I told you, nerve conduction, muscle contraction, everything. Then in the human cell, energy production is through aerobic respiration in the mitochondria. Energy production is in the mitochondria in the presence of oxygen. And there are different parts of complex tissues and organ system is there in a human cells. So these are the functional difference you can see in a microbial and the human cell. Can you just read it once?
Yeah.
If there is any doubt, just read and see if there is any doubt.
No, mom, I'm getting you. I will have to go through again and understand better. I'm getting you. It's okay.
Is it clear?
Yeah, it's clear.
See what is these help that for the antibiotics and probiotics. That is how helpful in the understood micro the production help this how various like for example COVID these are caused by different microbes. Antibiotic resistance, like use of any antibiotic, can also lead to resistant microbes. So these are the diseases causing problems. What will be affect the microbes? So one thing, microbes will cause different kinds of diseases, like viruses causing COVID-19 or bacteria causing pneumonia, and overuse of this antibiotic causes resistance in the body. Whereas in the health, these microbes are useful in the digestion, immunity. It helps to produce antibiotics, different kinds of vaccines, and probiotics. So that is how microbes has a role in health and in the disease. Is this clear? Is this point clear?
Yes, it's clear.
Next, why it is important? Why understanding of microbiology is important and how that can help a nurse. So one thing, when you learn about different microorganisms, it will help you to prevent and control the infection. You will get to know which microbe is causing this infection. So you can prevent and you can control the different types of infections. Second thing, you will be able to recognize what are the signs of that microbial infection. When you learn about different kinds of microorganisms, it will help you to understand what infection they will cause, what will be the signs and symptoms you can see in such patients. Second thing, it will help you to administer the appropriate antibiotics and other treatments effectively. When you know the microorganisms, you know what to be given, which drugs to be given for the patient, for the treatment. Then, being a nurse, you can educate your patient about the importance of hand hygiene and what are the preventive care they should follow. Then, microbials are also critical for the biotechnology, like for the production of antibiotics, for the fermentation, for genetic engineering. For that, also, we require the microbial cells. Then, in the environment, for the decomposition of waste, for the nitrogen fixation in the environment, we also require microbial cells. Then human health, like gut, inside our body also there are microbes, right? Good microbes are there, which will improve the gut microbe, disease causing probiotics, everything is required. And in the industrial application, like for the food production, for the production of biofuels, everything we require the microorganisms. So that is how microbes will play an important role in the day-to-day life. Is this clear?
Yes, ma'am.
Now we'll move on to the bacterial cells. Okay, each one we are going to discuss in detail. So what do you mean by bacteria? Bacteria are the single celled microorganisms which will lack a nuclear membrane and are metabolically active and divide by the binary fission, which is asexual reproduction. So this is how a bacterial cell will look like. So they are single celled organism and they don't have any proper nucleus membrane also. So they have a tail-like structure which is called as flagella. They have plasmid where their DNA is present, and then they have a plasma membrane, cell wall, a capsule which is outer covering, and the ribosomes are present, and they have pili or cytoplasms. So this is how a bacterial cell will look like. Understand what is bacteria? They are single-celled microorganism which lack a nuclear membrane and they metabolically active and divide by the binary fission. Binary fission means a single body will be divided into two cells and they replicate. Is it clear what is bacterial cell is clear?
Yes, ma'am, it's clear.
Yeah. Now we'll see what are the general features present in a bacterial cell. So usually bacterial cells are prokaryotic. Prokaryotic with means there is no nucleus present in the inside the cell and there is no organelles also present. That is why bacterial cells are classified as prokaryotic with no nucleus and no organelles. And they are always unicellular and there are different shapes are available like cocci, bacilli, spiral, vibrio. As you see in this image, there are different types or shapes are available for the bacterial cells. Different circular, rod shape, curved, other shapes. So these are various types of bacterial cells and their size typically ranges from zero point 0.2 to 2.0 micron in the diameter. So these are the general features you can see in a bacterial cell. So they are prokaryotic with no nucleus and organelles. They are unicellular and they are available in different shapes like cocci, bacilli and spiral, vibrio, and the size will vary from 0.2 to 2.0 diameter. Understood the general features? Understood the general features of a bacterial cell? Is it clear? Now, if you classify a bacterial cell based on shape, you can classify them into cocci, bacilli, and spiral based on the gram staining. Like there is staining methods. To study a microbe, you do stainings. Okay, so there are gram positive and gram negative stainings are there. And based on the oxygen requirement, you can classify the bacterial cell into aerobic and anaerobic. Understood? Understood? Understood?
Yes, ma'am, understood.
Now we'll see the structure of a bacterial cell. What all things are present in a bacterial cell? Okay. So usually bacterial cell has got a cell envelope. Cell envelope means a outer covering. So they are usually glycolactic. Glycolactic capsule or a kind layer. There is outside of a bacterial cell, there will be a capsule or a slime kind layer will be present. Now, what is this capsule? Capsule will be a thick and a organized and a firmly attached to the cell wall. So, every bacterial cell has got a cell wall. On the outer layer of the cell wall, there has a capsule, which is a very thick and a organized layer which is attached to the cell wall for its protection, or it has got a slime layer. Slime layer will be a thin, unorganized and a loosely attached layer for its protection. Now, what is the function of that capsule or slime layer? It will provide protection from the discrete immune evasion and the attachment to the surface. So, mainly your bacterial cell has got a cell envelope. The cell envelope is an outer covering. The outer covering can be either a capsule layer, which is a very thick layer, or it can be a slime layer, which is a very thin layer. Now, what is the function of this capsule or a slime layer is? It provides the protection for the bacterial cell. Is it clear? Cell envelope is clear?
Yes, it's clear.
Next, cell wall will provide the structural support and shape for the bacterial cell. Okay, inside that you have a cell wall which will provide the structural support and shape for your bacterial cell. And they are composed of peptidoglycan. And this peptidoglycan will become thicker in the gram positive bacteria and thinner in the gram negative bacteria with an outer membrane. That is, I told you to study the microorganism, you stain them. You stain with the different colors. So the cell wall will get thicker when you use a gram positive staining and becomes thin when you use a gram negative staining. So this staining you will learn in the upcoming slides. Okay. So second structure we can see is a cell wall which will provide a structural and functional support for the bacterial cell. Then you have a plasma membrane, which will control the material exchange. So this is a phospholipid bilayer, which has got embedded all the proteins in it. And what is the function you can see in a plasma membrane is selective permeability, transport of nutrients and waste materials, site of metabolic processes like respiration. So everything, exchange of materials will be happening inside this plasma membrane layer. Understood what is plasma membrane? It is a layer, inside layer where the different exchange will be happening. Exchange of nutrients, waste products, selective permeability, all these things will be happening inside the plasma membrane layer of a bacterial cell. Is it clear what is cell wall and what is plasma membrane? Is it clear? Is this clear?
Yes, it's clear, Mom.
Next one is cytoplasm. Cytoplasm is a gel-like substance which you can see inside the cell membrane. And that cytoplasm will contain different enzymes, nutrients, organelles, and the microcellular essential for the cellular process. So this is another structure you can see inside a bacterial cell, cytoplasm, which is a gel-like structure which will contain different enzymes, nutrients, and organelles for which is required for the cellular process. Okay.
Okay.
Next is genetic materials. Genetic materials will include your nucleotide, plasmid, DNA, RNA, everything. So nucleotide is a region which contain the bacterial chromosomes and they are not enclosed by any sort of membrane. Whereas plasmids are a small circular extra chromosomal DNA which will carry the genes for the antibiotic resistance and other traits. Then it also contains ribosomes for protein synthesis and they are always smaller than that of a eukaryotic ribosome. So this is another structure you can see in a bacterial cell. Genetic material which will be present inside the nucleotide and plasmids are the small cellular extra chromosomal DNA which will be carrying all the different types of genes and ribosomes also present for the protein synthesis. Clear?
Yeah, yes, Mom, yes.
Next one you can see is the appendages. Appendages means flagella, cilia, pili, etc. Like hair kind structures you can see in a bacterial cell. These are called as appendages. So first one you can see is a flagella. Flagella is nothing but a long whip-like structure for the motility. For the movement of the bacterial cell, they have flagella. And there are different types are based on the arrangement, like how the flagella is been arranged. You can classify them into monotrichous. Monotrichous means only a single flagella is present for the movement. It is called as monotrichous. When there are flagella is present at the one pole, only one side if it is present, it is called as lophotrichous. When in the both the sides, if the flagella is present, it is amphitrichous. And in the all over the body, if the flagella is present, it is petritrichous, as you see in this image. If the whole body is having the flagella, it is petritrichous. Understood? So this kind of structure you see is the flagella and the cilia. Sorry, can you see the slide?
Yes, Mom.
Yeah. Now they have pili. Pili is a hair-like structures which help them in the attachment to the surfaces. So this hair kind structure you can see, they are called as the pili, which will help in the attachment to the surfaces. And this is the flagella, which helps in the movement of a bacterial cell. Hair kind structure is called as pili, well, which helps in the attachment of this bacterial cell to the surfaces. Now they have sex pillars, which is a specialized structure for the conjugation. Conjugation means for the transfer of the genetic materials. Understood the appendages? Understood?
Yes, understood.
Yeah. The last one you can see is that endospores. Endospores, like some bacteria will produce this endospores under an unfavorable condition. Like to sustain this bacteria, they produce endospores, which is resistant to heat or which is resistant to any dissociation or any chemical or any radiation. When the bacterial cell has to survive, they produce endospores in order to sustain themselves under the unfavorable condition. For function of this endospore is for the survival mechanism. Okay, and as I told you, there are different types of bacterial cells are there: cocci, bacilli, and spiral. These are different types of shapes: spherical, rod shape, and spiral shape. These are different types of shapes of bacterial cells are available. So, understood the structure of a bacterial cell? Understood the structure of a bacterial cell?
Yes, mom, yes.
Yeah. Now we'll see the function and the importance of this bacterial cell. One is in the ecological role, like this microbial cells will helps in the decomposing of the different organic matters, and for the cycles, different cycles like carbon cycle, nitrogen cycle, for the synthesis of different things, these microbes are essential. Second thing, medical significance, like for a cause of any infection or for the source of any antibiotics, microbes are very important. For the industrial application, like fermentation of your yogurt, production of cheese, alcohol, and any other bioremediation to clean up the soil, pollutants, everything, microbial activity is important. So that is why microbes are important in the day-to-day ecosystem. For the decomposition of organic materials, study of any infection, for the production of any antibiotics, or for the production of yogurt, cheese, alcohol, all those things, microbes are very important. Clear?
Yes, mom.
Yeah. Next we'll see about the virus. Okay. So virus is a typically described as a obligate intracellular parasite. Viruses are called as parasite because they can't live alone. They have to get into someone's body and then they multiply. For example, COVID-19, it affected different people, right? And they multiplied. So that is why they call it as parasite. So they are acellular infectious agent which require a presence of a host cell in order to multiply. So the host cell will be the human body. So into the human body, this infectious agent will enter and they will multiply. The virus has been found to infect all the types of cells, including human cell, animal cell, plant cell, bacteria, yeast, archaea, protozoa. So virus can infect all types of cells. Understood what is virus? Understood?
Yes, yes. Yes, mom.
Now we'll see what is the structure of a virus. What all things are present in a virus. Okay. So one thing you can see is a protein coat. A protein coat will be seen in the virus, which is called as a capsid, and there is a genetic materials present like DNA and RNA are also present. So some will have an envelope, which is a derived from the host cellular membrane. And why do they have all this is to obligate the intracellular parasite. For example, in case of influenza, HIV, COVID-19 viruses, you can see a capsid. Capsid means a outer protein coat layer, and they have genetic materials like DNA and RNA. And they have an envelope which will cover the outer layer of the virus. Now we'll see what is capsid in detail. So capsid is nothing but a protein coat. So function of this capsid layer is it will protect the viral's genetic materials and aids the virus ability to infect a host. So as I told you, it is a protein layer inside which the genetic material like DNA and RNA will be present, and that will be protected by this capsid layer. Now the structure is just, if you see, the capsid layers are made up of different protein units called as capsomeres. So these capsid layers are made up of capsomeres, and this capsid can have different shapes such as helical shape, isosecondary shape, or complex shape. For example, in this image you can see this is how a virus structure will look like, which has got an outer envelope and inside they have a viral genome present. Understood? So the outer layering of a virus is called as a capsid layer, which contain or which protect the genetic materials. And they are capsids are made up of capsomeres and they are of different shapes such as helical shape, isosecondary shape or complex shape. Clear protein coat capsid is clear.
Yes, ma'am, clear.
Next is genetic material. Genetic materials like the genome will carry the information required for the virus to replicate and to assemble the new viral particles. And the different genetic materials are DNA and the RNA. These are the different types of genetic materials you can see. So DNA are usually double-stranded DNAs and RNAs are usually the single-stranded RNA. So these are the different genomes you can see inside a virus. Now, where exactly it is located? This genetic material is housed inside a capsid layer. So, inside this capsid or the protein layer, you can see the genetic materials. And in some virus, they may have directly surrounded by a envelope. Understood where the genetic material is present inside a virus? So, inside the virus body, there is a capsid layer which is made up of capsomeres. Inside that capsid, the genetic materials will be preserved. Clear?
Yes, ma'am, clear.
Yeah. They have something called as envelope. Envelope is nothing but the outermost layer of a virus. So the function of the envelope is a lipid bilayer derived from the host cell membrane and they serve as an additional protective layer. Outside this capsid layer, there is an envelope. It is optional. Some viruses may have and some virus may not have. So it is actually another protective layer for the virus. Now the structure, the envelope will contain viral glycoproteins which are essential for the virus to attach to the host cell receptors. Now the presence, as I told you, not all the viruses will have the envelope. Those who do not have typically referred as envelope viruses and they do not have this one is non-enveloped viruses. So all the viruses won't have the envelope layer. It is an additional protective layer. If that envelope layer is presented, then it is called enveloped viruses. If that layer is absent, they are called as non-enveloped viruses. Understood? Understood the envelope layer, the outermost layer?
Yes, ma'am, understood. Understood.
How the structure of a virus will look like. As I told you, you can see the envelope layer, the outermost layer. And then inside they have a capsid layer. And inside the capsid layer, the genetic materials will be preserved. Clear?
Clear, mom, clear.
Next is various classifications of the viruses. If you see, there are different classifications. First thing, by the genetic materials only, you can classify them into DNA and RNA viruses. Then based on the shape, as I told you, there are different types of shapes you can see. Helical, isosecondary, complex. So there are different types of viruses based on the shapes are also present. So these are the different viruses present. Influenza virus, hepatitis virus, adenovirus, rotavirus. So these are different types of viruses present. So in this, I have given the difference between a bacteria and a virus and what is common in both. Can you just read? Can you see the slide?
Yes, mom.
Read and see if there is any doubt between bacteria and between the virus. Just read and see if there is any doubt.
No, there's no doubt. I'm following you.
Okay. Next one we're going to learn is about the fungi. Okay, so we finished bacteria and virus. Next is fungi. So fungi are a group of eukaryotic organisms that includes yeast, mold, and mushrooms. So these things will come under the fungi category. Now, if you see the structure of a fungi, they are eukaryotic cells. Eukaryotic cells means which has got a defined nucleus and organelles are present. So fungi is a eukaryotic cell which contain a chitin cell wall. So chitin cell wall is the outer layer of a fungi you can see. Okay. Now different types of fungi are present. As I told you, yeast which is a unicellular, mold which is a multicellular. So different types of fungi are also present. Now, why it is important? One is for the production of any antibiotics like in penicillin or for the production of any fermentation, you require the fungi production. And harmful cause of the fungi is it can cause various infections like Candida species, Candida aspergillus. Various infections can also be caused by the fungi. That is the importance. One is beneficial importance for the production of any antibiotics like penicillin or for the fermentation. Harmful effect of fungi is it can cause infections like Canadian aspergillus can cause various infections in the human body. Understood what is a fungi? So they are a eukaryotic systems with a
Yes, ma'am. Understood.
Now, if you see the classification of a fungi, you can classify them by the structure like unicellular or a multicellular. Unicellular example is the yeast, and multicellular fungi can be either molds or mushrooms. Then you can classify the fungi based on the reproduction method. Reproduction method you can see asexual as well as sexual reproduction you can see in a fungi. Asexual by the production of spores. By the spores formation, they will multiply, which is a asexual reproduction. Next, by fusion of specialized cells, they reproduce by the sexual. So these are the different classification of a fungi. By structure, they can classify it into unicellular and multicellular, where the way they reproduce themselves is asexual or sexual reproduction can occur. Is that clear? Classification is clear?
Yes, ma'am. It's clear.
Okay. What are the different components you can see in a fungi? Okay. For example, see, this is how a fungi structure will look like. Okay. So this is how a fungi structure will look like. And what are the different things you can see in a fungi? One thing you can see is a hyphae. Hyphae is nothing but a basic structural unit of a multicellular fungi. They look like a long thread-like structure which will grow and spread to form a mycelium. As you see in the image, see the root portion you can see like a hair kind structure. Can you see? This is what we call as
Yes, I can see.
this is what we call which will be like a thin, long hair-like structure which will grow to connect with the mycelium. Understood? So this is the first structure you can see in a fungi. So hyphae is a long thread-like structure that will grow and spread to form the mycelium. Now what is mycelium? It is a network of hyphae which will act as a vegetative part of the fungi, and they are responsible for the nutrient absorption. So from the soil, the nutrients has to be absorbed, right? So this hyphae will grow and form the mycelium, and that will take up all the nutrients for the growth. Understood what is hyphae and mycelium?
Yes, ma'am.
Yeah. Next is a cell wall. As I told you, it is composed of a chitin, which is a outermost layer, which will provide the rigidity and protection for a fungi. So it has got a cell wall for its rigidity and for the protection. Now the fungi will produce spores for the reproduction. So this will help the fungi to spread and propagate. And this can be dispersed by the presence of wind, water, or any animals. So these are the spores they produce, which will be spreaded and multiplication of the fungi will be happening. Next one is a fruit body. That is some specialized structures which contain spores. For example, mushrooms, which will release the spores for the reproduction. Those things we call as fruit bodies. So as you see, In the image, you can see the green color thing which will contain the spores inside. So that is called as fruit body, which will have spores which will help in the reproduction. Understood? Understood what are the components of a fungi? What are the different components of a fungi is clear?
Clear, very clear.
One is hyphae, one is mycelium, one is a cell wall, spores, and a fruit bodies. Now we'll see some of the fungi which will commonly cause infections or in the immune compromised patients. Those patients who have less immune power, in those patients, this fungi will cause various infections. So we'll see one by one each. Okay. What are the different infection causing fungi? What are the species? The very first thing is the Candida species or Candidiasis. So this is one of the fungi species which causes infections in the human body, especially in the immune compromised patients. This Candida species, which is a species which comes under the fungi, which causes the infection. Now we'll see what is the causative agent is Candida albicans. So this is one of the major causative agent which can cause infections like oral candidiasis. Oral candidiasis means like white color patches will form inside the mouth and the throat. Then esophageal candidiasis, like in the esophageal region, there will be infection which can lead to pain and swelling. Next one, vaginal candidiasis. Vaginal candidiasis means which is very common in the woman, which can cause itching and discharge. Last one is invasive candidiasis. This is a serious infection that can spread into the bloodstream, which can affect the heart, kidneys, and the other organs. So this is the one of the fungi species. This is a causative agent, and these are the infections occurring. So for the example, in your exam, if a question is asked, list out the various fungi species which can cause various problems in immune compromised patients. You can write one is Candida species, causative agent is Candida albicans, infections caused are oral candidiasis, esophageal candidiasis, vaginal, and the invasive candidiasis. Understood?
Yes, ma'am.
Yeah. So this is how the infection will look like. Okay. Next species is Aspergillus species. It is another variant of your fungi. Here the causative agent is Aspergillus fumigatus, which is a very common species of a fungi. Now what are the infection they causes? Your pulmonary aspergillus, like infections of your lungs can be present, like pneumonia like symptoms will be present. So they will affect your lungs. Second thing is invasive aspergillus, which will often affect the lungs, sinus, brain, and other organs, which will lead to the damage of tissues. Next one is allergic bronchopulmonary aspergillus. Again, in the patients who are having asthma, they or any fibrosis, which will cause in the patients. So this is a common species of a fungi, Aspergillus species. Here the causative agent is Aspergillus fumigatus. Infections caused are pulmonary aspergillus, like pneumonia-like symptoms will be there. Invasive aspergillus, again affecting your lungs, brains, and other organs, which can cause tissue damage. Allergic bronchopulmonary aspergillus, which can affect the in patients like asthma and leads to cystic fibrosis in the patients. Is it clear? Second species is clear?
Yes, ma'am, yes.
Yeah. So this is how this aspergillus will affect the lungs. Because of this, the spores will be inhaled. By mistakenly, it will be inhaled by the human body, and that will go and affect your lungs. And the lung has got lumens, like the inner layers will be damaged and causes various lung problems. Next species is Cryptococcus. So Cryptococcus species is another common causative agent. Here it is Cryptococcus neoformans. So this is the major disease causing agent. Now what are the infection they causes? One is meningitis. Meningitis is nothing but the inflammation of the brain as well as spinal cord, and which can cause severe headache, nausea, and the confusion. Second thing they cause is pulmonary cryptococcosis, which can affect the lungs and cause pneumonia-like symptoms, especially in the patients who are having HIV, AIDS, and everything. Another one they cause is disseminated diseases. Like the infection can spread to different organs, especially liver, kidneys are affected. Understood? The species is Cryptococcus species. The disease-causing agent is Cryptococcus neoformans. Infection they causes, one is meningitis, which is an inflammation of brain and spinal cord. Second thing is pulmonary cryptococcosis, which can cause pneumonia-like symptoms in the patients who are having HIV or AIDS. Or it can cause disseminated diseases, which is infection affecting the different organs like liver, kidneys, etc. Understood?
Okay, ma'am.
This is how it will affect the
Yes, ma'am.
brain, lungs, and everything. It causes inflammation in the brain, spinal cord, and the lungs are also affected. Next one is pneumocytosis jirovecii. So there the common causative agent is pneumocystis jirovecii. Infection they causes again pneumocystis pneumonia, which is a serious fungal infection affecting the lung, which can lead to severe respiratory distress. Another species is mucorales. Mucorales is another causative agent. The causative agent is mucor, rhizopus, and other mucoral species. Like various species will affect the human body. This causes infections like rhinocerebral mucormycosis. That is infections of your sinus, your brain, which will be commonly seen in the patients with a diabetic ketoacidosis. Another infection they cause is pulmonary mucormycosis. That is infection affecting the lungs. Then in the post-transplant patients. And the another infection they affect is cutaneous mucormycosis. That is the infection which is affecting the skin, especially particularly in the trauma victims. So these are the another infections which will be causing by the fungal species. Is it clear?
Yes, ma'am.
Yeah, ma'am. Clear.
So this is how they affect the lung regions. Next species is Histoplasma capsulatum. Histoplasma capsulatum, here the causative agent is Histoplasma capsulatum. Infection they causes again the pulmonary histoplasmosis, which is causing the pneumonia-like symptoms, and disseminated histoplasmosis, which can spread into your bloodstream. It can affect your liver, spleen, and the other organs. So again, it been spread to the through the soil or by consuming anything which has got that infection, and that will be entering the human body and affect the human body's lungs. Next one is blastomyces dermatitis. So here the causative agent is blastomyces dermatitis. Infection they causes pulmonary blastomocytosis, like respiratory symptoms like cough, fever, chest pain, and disseminated blastomocytosis, like it can affect the skin, bones, and the central nervous system also. So these are the various infections caused by your fungi. Is it clear?
Yes, ma'am.
So parasites.
Yes.
Okay. So what is parasites? They are also eukaryotic organisms which can live on or in a or host organ. So they live in or within a host organ. Parasites will be living, and they are also eukaryotic organism with a defined nucleus and the organelles present. Now, there are different types of parasites are present. Protozoa, which is a single cell, helminthes, which is a multicellular worms, ectoparasites, which lives in the host surface. And these various parasites will cause severe diseases and drain the nutrients from the host body. Especially these parasites are affecting the human body and then drain the human body also. So these are the parasites which we'll be discussing in detail. Understand what is parasites? They are eukaryotic organisms which will live on or in a host organism. There are different types of parasites. One is protozoa, helminthes, and ectoparasites. Effectors, they will they will live in the host body and they will drain the nutrients from the host body. Clear? Parasites are clear?
Yes, ma'am.
Yeah. Now we'll see each one in detail. So first one, if you see protozoa, protozoas are a single cell parasite. As you see in the image, they are a single cell parasites. So definition is protozoas are unicellular eukaryotic organism which can cause a wide range of disease in the human body. Now, protozoa has got a life cycle which will involve one or more hosts. And this can be either complex with different stages like trophocytes, which is an active stage, and a cyst formation, which is an inactive state. So this is how the protozoan life cycle will take place. So this protozoa can live in one or multiple host body, and they have different stages. One is active mode and another one is inactive mode. So that is how their life cycle will take place. Clear? Clear?
Yes,
Yes,
yes, ma'am.
mother. Clear.
Yeah. Now we'll see what is a common protozoan parasite with a life cycle. So which is a very familiar, I think, malaria, spread of malaria. Okay. So this malaria has been caused by Plasmodium species. Okay. So this is a very important thing. So please listen. So malaria, which is caused by Plasmodium species, and now their complex life cycle will involve humans as well as mosquitoes. So these are the major things which is involved in their life cycle. Now how this infection spread is, infection is spread via the Anopheles mosquitoes. Anophele mosquitoes bite the human body. So when the Anophele mosquito bites, what will happen is this mosquito will have the sporocytes inside the body, which is actually carrying the infection. So when they bite the human body, these sporocytes will enter the human body, especially in the liver. So in the liver, the sporocytes will go. And in the liver, the sporocytes will try to multiply. And that will be released into the bloodstream. And in the bloodstream, in the RBCs, they will again multiply and they form eggs. And that will be circulated in the human body. When another mosquito comes and bite, that mosquito will get infected and that life cycle will keep on moving. And the different stages which is happening inside the human body is first is sporocytes. When the infected mosquito bite, the first thing is sporocyte, that is passage of this infection into the human body. Next one is merocytes. Merocytes means inside the liver, they will multiply and they is released to the human body's bloodstream. Next is gametocytes. Inside the RBC, again they multiply and they form cells. That is what gametocytes. And then once an unaffected mosquito come and bite that human, that is passed on to the mosquito and then the life cycle will continue. Understood how the malaria has been caused?
Yes, mom. Yes.
Is the life cycle clear? As you see in the image, if you see in the image, it is mentioned. See, the first thing is transmission to the human body, that is the sporocyte stage. Second is sporocyte will enter the liver and cause infections. Then in the liver, they will start to rupture and multiply. That is called as merocytes. And then they are released into the human bloodstream and their sexual cycle will start. And that is gametocyte. And then it is again transmitted into the mosquito. Now is it clear?
Yes, mom. Clear.
Yeah. Fine. Now next species you can see in the protozoa is Leishmanian species. So Leishmanian species, here the life cycle will involve sand flies. There it was mosquito, here it will be a fly. So fly will be act as a vector and affect the human body as a host. So here what happens is, again there are different stages like amastigoid, which is happening inside the host cell, and promastigocyte, which happens in the sandfly. So here what is happening is the sandfly will be infected. Okay, can you see the image? Can you see the image? So your sandfly will be infected. They will bite the human body. And when they bite, they release the infective materials into the human. Human body, what it has got is, human body has macrophage, which will engulf any sort of antibodies, right? So your microphages will engulf that infections, and that infection will try to grow inside the host body, that is especially inside your macrophage, and then that will be multiplied and that will be released into the bloodstream, and again, and that unaffected sandfly will come and bite the human body. The infection will be passed on to the sandflies, and that they will be multiplied, and that will be again causing the life cycle to occur. So this is what Leishman's species causing infection. Is this clear?
Yes, ma'am.
Is it clear?
Yes, it's clear.
Yeah. Next one, toxoplasmosis. Again, another species which has got life cycle, which includes primary host, which is felines, and an intermediate host like humans. And here they will shed their oocytes, and then they will first have a techucyte. Techucyte means a highly active site. Then they have a low active site, that is bradiocyte stages. For example, here the infection, how it is spread is uh, the soil also, or soil will have the infections, and in the soil, whatever plants are growing, when the human body consume the same, that will be passed on to the human body. Or any cattle, they are having the same grass or something which is grown in the same infected mud, the cattle also will carry on this infection, and this cattle will be consumed by the human body. So again, there is a passage of infection into the human body, and then once it enters the human body, there is tachyzoid. Tachyzoid is the first stage. Tachyzoid is the first stage where is an active multiplication of the organism will be seen. Then it affects the tissues of the body, and then there will be bradiozoid. Bradiozoid means low activation of the infective cells will happen, and that is how the infection will be passed on. So that is how toxoplasmosis will infect the human body. Is that clear? Is it
Yes,
clear?
ma'am. Yes.
Yeah. Now the second which comes under the parasite is helminthus. So helminthus is another multicellular parasite, which is a parasitic worms which can live in or in the host, typically having a complex life cycle with a multiple host. So their life cycle will often involve a definitive host where the adult forms will live, and the intermediate host where the larva or the juvenile forms will develop. So that is how they will complete their life cycle. So one adult will form, they lay their eggs, then the larva will be formed, again they will grow. So this is how the helminthus worms life cycle will be going on. Understood? So they often have a definitive host cycle where the adult forms will be living. Then they have an intermediate host where they lay the eggs and larva will be developed. So that is how the helminthus life cycle will be going on. Clear?
Yes, clear.
So these are different types of helminths you can see: roundworms, flukes, tapeworms, etc. Understood? These are the different types of helminths you can see. These are present inside the human bodies also. Clear?
Yes, ma'am.
So this is how the infection is passed on in the human body. Like the cattles and all usually have this types of helminths or worms are present in the cattles. And when these cattles are eaten by the human body, and if they don't cook the meat properly, what will happen is the same hookworm or tapeworm will enter the human intestine, and there it will start to multiply, and like that the life cycle will goes on. Clear? So cattle, they will consume this worms, and if the meat is not cooked and eaten by a human body, this will be passed on to the human intestine, and there they will try to multiply. Understood? Understood?
Understood.
Yeah.
Yes, ma'am.
What are the different common helminths parasites with their life cycle? So very first thing is a Stichomasis, systematic species which will involve a human as a definitive host and snails as an intermediate host. So their life cycle, how it will be is, first the snails will lay their eggs, then that eggs will form the larvae, and then the snail will be born, and then they start to infect the human body. Next one is liver flukes. Liver flukes also, there is a definitive host is a human or a liver stock, and they have an intermediate host as a in the freshwater snails. So how their life cycle will be is, first they will lay their eggs in the water, and then the larva will be infects the snails in the water, then they will infect the aquatic plants. So that is how the liver flukes life cycle will be going on. Is this clear?
Yes, ma'am.
Yeah. So that is what I have explained here. They will lay the eggs, then that will infect the snails, and then they will infect the aquatic plants, and then it will infect the human body. Next one is tapeworms. Tapeworms are here also the definitive host will be human, where the adult forms will be living in the human intestine, and the intermediate host will be the pigs or the cattle. As I told you, the pigs and cattle will have this tapeworms in their body. So if it is not properly cooked and if it is consumed by the human, they will also get the same thing. So here the life cycle will be, first they will lay their eggs and they form larvae in the muscles and the indigested by, it will happen in the cattle or in the pigs. And when that is ingested by the human, the adult tapeworms will be start to develop in the human intestine. And that is how the tapeworms life cycle will be continued. Is it clear?
Yes, ma'am.
Next we see life cycles involving multiple hosts. Till now you have seen only two hosts. In some parasites, there are multiple hosts are present. For example, there will be a definitive host. Definitive host means a host where the adult or the sexual stage of the parasite will be developing. So the host where the adult form is developing is called as a definitive host. Second one is the intermediate host, where they will form the larva or where they lay the eggs and that larva will be formed. Third one is parasitic host. It is a host that will carry this parasite but do not contribute to the life cycle of a parasite. So this is the different hosts involved. One is definitive host where the adult form will live. Second is the intermediate host where they lay the eggs and larva will be formed. And the last one is parentic host, which will do not have any contribution to the parasite's life cycle. For example, if you see examples of parasites with a multiple host life cycle is, one is malaria. It has got mosquito, it's a vector, and the human body will be the definitive host. Second thing is sociomycetes, where there is a snail is the intermediate host and human will be the definitive host. Last one is taper, where the cattle and pigs will be the intermediate host and humans will be the definitive host. Is it clear?
Yes, ma'am.
Yeah. Next one, see the same thing I've been explained here also, the malarial life cycle, as we have discussed how the spread will be occurring. Just go through this life cycle and see if there is any doubt. Just go through the cycle. Is there any doubt?
No, ma'am, no.
Clear?
Clear.
Yeah. Next one is the cystomeiosis life cycle between the snail as an intermediate host and human. So just read it once and see if there is any doubt. The first one,
Okay.
then that will develop, affect the snails, then they multiply, then they enter the human body. Inside the human body, they will further grow. Any doubt?
Not at all.
Yeah. Next one is the tapeworm cycle. As I told you, cattle or the pigs will be infected. Then in their intestine, it will be growing. When that meat is consumed by the human, in the human intestine, this will further develops. Is it clear?
Yes, ma'am.
Next one is organic molecules in the microbial system. So I hope you have learned the biochemistry last class, okay? So organic molecules are the one which has carbon-based components, which is essential for the life and are critical in the structure, function, and the metabolism of any microorganisms. Now, what is their importance in the microbial system is, one is to provide energy to support their structure and for the genetic information. So supporting microbial growth, supporting microbial reproduction, adaptation to the different environmental changes, etc., requires different kinds of organic molecules. For example, of organic molecules includes carbohydrates, lipids, proteins, and nucleic acids. So these which develops the mix of the organic molecules. I hope you have learned about carbohydrates, lipids, proteins, and nucleic acids, right? Yes or no? Yes or
Yes,
no?
ma'am.
Biochemistry class you have learned, right?
Yes.
Yeah. Now we'll see one by one in detail. So one is carbohydrate, right? So what is carbohydrate? So carbohydrates are the one, they are organic compounds which are made up of carbon, hydrogen, oxygen, and they are often in the form of sugars and starches, right? So carbohydrates are organic compounds which are made up of carbon, hydrogen, oxygen, and they are often in the form of sugars and starch. Now, where there are different types of carbohydrates are available, right? Monosaccharides, disaccharides, and polysaccharides. So monosaccharides are the simplest form of sugar, for example, glucose and fructose. Disaccharides are two monosaccharides joined together to form disaccharides. For example, lactose and sucrose. And polysaccharides are the one which forms a long chains of monosaccharides. Example, starch, glycogen, and cellulose. Yes or no? Yes or no? These are the different types of carbohydrates, right? Understood what is
Yeah.
carbohydrate? What are the different types of
Yes.
carbohydrates here?
Clear.
Yeah. Now, what is the function in the microbial system? One is for the production of energy source, like glucose for the cellular respiration, then for structural components like peptidoglycan in the bacterial cell wall, cellulose in the plant, for the energy storage like glycogen and starch is required in the microbes. That is why these kind of carbohydrates are important in the microbial system. For example, for the lactobacillus use lactose for the fermentation, right? So there you can see the application of lactose. Now for the Streptococcus pneumonia, they has a polysaccharide capsule for the protection. So everywhere in the microbe you can see there is organic molecules associated with the same. Clear? So for a
Yes.
for the energy source, for the components, structural components, and for the energy storage, they require carbon. Carbohydrates are required. Clear?
Clear, mom. Clear.
Next one. Drug development strategies. Usually the glycan-based vaccines have been produced with these microbes. Like there will be conjugating of these carbohydrates with the proteins to elicit strong immune responses. Like they will transfer these carbohydrates along with the proteins to prepare a strong immune responses, to prepare vaccines. For example, vaccine against meningitis or vaccine against pneumonia, they create by mixing of carbohydrates and proteins together. That's how they create different drugs, like vaccines are produced. Understood drug development strategy? Understood?
Yes, ma'am.
Yeah. Next is targeting carbohydrates in drug development. Like when there is a drug development is done, there will be inhibit of cell wall synthesis. Like only the targeted peptoglycan will undergo the synthesis in the bacteria, and rest everything will be protected by the cell wall. So only the targeted peptidoglycan or targeted layer only will undergo the synthesis in the drug development. For example, in the production of antibiotics like penicillin, only the targeted layer will undergo the synthesis and the vaccine will be produced. Second thing is disrupting biofilms. Again, here also, again, the targeting extracellular polysaccharides will be undergo the synthesis and the rest will prevent the biofilm formation in the pathogenic bacteria. So whenever the drug development is done, only the targeting cells will undergo the synthesis and rest will be protected. Clear?
Yes, ma'am.
Next is lipids. There's another structure and the lipid structure and function you will see. So lipids are another hydrophobic organic molecules which will consist mainly of carbon, hydrogen and oxygen, and they are not soluble in the water but may be soluble in any other non-polar solid. So they are called as lipids. Now there are different types of lipids are also available. For example, fatty acids and triglycerides, like fatty acid chains attached to the glycerol. Or there are phospholipids available, which are essential for the membrane structure and with a hydrophobic head and a hydrophobic tails. That is why they are insoluble in the water. Then there are another type of lipid steroids, which will include cholesterol, which is involved in the membrane stability. So these are the different types of lipids you can see. Understood? So lipids are a hydrophobic organic molecule which contains mainly of carbon, hydrogen and oxygen, and they are not soluble in water. And there are different types of lipids. One is fatty acids and triglycerides, one is phospholipids, and one is steroids. Is it clear, lipids and types?
Yes, ma'am.
Yeah. Next, what are the function of lipids in a microbial system? Again, for the energy storage, for the membrane structure to be maintained, and for signaling molecules, your lipids are important. That is energy storage. The triglycerides will store the energy in the microbial cell. That is how lipids will help. Second thing, membrane structure. Like the phospholipids will form the bilayers in the plasma membrane and protect the membrane structure. And the last one is signaling molecules like the steroids and the fatty acids will play an important role in the microbial signaling. That is why lipids are important in the microbial system. For example, if you see Mycobacterium tuberculosis, they have a lipid-rich cell wall which will make it resistant for the antibiotics to attack them. And the another one is Escherichia coli. They have a cell membrane which is composed of phospholipids. So it has a membrane structure they will get. Understood why lipids are important in the microbes? One, for the resistance, and one for a membrane structure. They have the requirements. Clear what is the importance of lipids in the microbes?
Yes, ma'am.
I'll just read it once and see. Is it clear?
Yeah, it's clear.
Yeah. Now we'll move on to the targeting lipids in the drug development. Again, there are another drugs are also developed from the same, like inhibition of biosynthetic pathways. Like it will block the various enzymes and pathways which are involved in the lipid synthesis. For example, statins will inhibit the cholesterol synthesis. It will inhibit the cholesterol synthesis in your body. It will lower the cholesterol synthesis in the body and helps the production. Understood? The one
Yes,
example
ma'am.
in the targeting lipids in the drug development. Now, lipid as a drug target. For example, cholesterol, if you see, drugs like statins will target the exact enzymes in the cholesterol and that will undergo the biosynthesis to decrease the LDL. What is LDL? Low lipid cholesterols. Okay. So that will reduce the overall cardiovascular risk factors also. So in the cholesterol, the drugs like statins will only target which enzyme is required to be undergone the synthesis. And then it will lower the LDL level in the body and thereby it reduces the overall cardiovascular risk also. Another one is triglycerides. So fibrates, it's a classification drug class of our triglycerides. So here the fibrates and the omega-3 fatty acids derivatives the modulate triglyceride levels. That is, here this also will reduce the triglyceride level in the body by affecting the only the specific peroxium polyfer activated receptors. Only the specified receptors will be activated in order to reduce the level of the triglycerides in the body. So that is how a drug will be developed. Is it clear?
Yes, ma'am. Yes.
Next one is protein. So proteins are the large molecules which are made up of amino acids which are linked by the peptide bonds essential for a wide range of cellular functions. Now there are different types of proteins are available. For example, enzymes, which is a catalyst for a biochemical reactions. What do you mean by catalyst? Anything which will increase the rate of reaction. Okay, so there are enzymes which will improve the or increase the rate of reactions for a occurring. For example, amylase or lipase. Another type of protein is structural protein, which will provide a structural support for the microbes. Another one is transport proteins that will move the molecules across the membrane. And the last type is antibodies, which is a part of the human response. So these are the different types of proteins present in the body. Clear? What is proteins? They are the large molecules which are made up of amino acids. They are linked together by peptide bonds. And there are different types of proteins like enzymes, structural proteins, transport proteins, and antibodies. Is it clear?
Yes, ma'am.
Yeah. Now we'll see what are the functions of these proteins in a microbial system. One thing, it will catalyze the rate of reaction. So enzymes like DNA polymers and RNA polymers for the replication and transcription, this enzyme will increase the reaction. Second thing, for the cellular structure and movement, like as I told you, cytoskeleton proteins in the bacteria and eukaryotic microbes will provide the structural support and the helps in the movement. Third thing is transport and communication. Like membrane bounded proteins will facilitate the nutrient uptake and signaling transduction. For example, if you see E. coli enzymes like beta galactorase for the lactose metabolism and for this pneumodons arginus will use protein channels for the nutrient uptake. So that is how proteins will have a role in the microbial system. One, it will improve the rate of reaction. It will provide cellular structure and helps in the movement, helps in the transportation of different ions and helps in the communication and helps to facilitate nutrient uptake. Understood how the proteins help function in a microbial system?
Yes, ma'am. Yes.
Yeah. Next is targeting proteins in the drug development. Again, enzyme inhibition, like inhibiting only the bacterial enzymes like DNA or the ribosome protein for any production of drug. Only the specific bacterial enzyme will be undergoing the inhibition. Second thing, targeting the toxins, like neutralizes the bacterial exotoxin. Some bacteria will release the toxin outside the cells. So only that will be targeted and that will be neutralized. Last one is inhibiting protein synthesis, like drugs like chloramophenol inhibit only the protein synthesis by only the targeting the exact microbial ribosome. Rest everything will be left untouched. So only the targeting microbes will be undergoing the synthesis and helps in the production of drug development. Is it clear?
Yes, ma'am.
Next one, the last one is the nucleic acids. Nucleic acids is nothing but another macromolecules which will carry the genetic information and are made up of nucleotides like sugar, phosphate groups, and nitrogenous base. And there are different types of nucleic acids are present. That is DNA, deoxyribonucleic acid, which contain the genetic blueprints of the organisms, and they are double-stranded in helix. Whereas the another one is RNA, which is a ribonucleic acid, which is a single stranded, and it involves all the protein synthesis. And it has got mRNA, tRNA, and rRNA. Now, what are the functions of this nucleic acid in a microbial system is, one, it helps in the genetic information storage. As this contain DNA and RNA, which has got the genetic information, it helps in the storage of this genetic information. So DNA in the genome will carry all the instructions for the cell growth, cell function, and cell replication. Then it helps in the protein synthesis. RNA is the main place where the proteins will undergo the synthesis. So this is a key for translating the DNA into the proteins via transcription and translation. Transcription and translation, the process of how it undergo the synthesis. And then gene expression regulation, like microbes will regulate the genes in response to the environmental condition, like regarding the how the environment it is, under that state, the gene expression will be manipulated or regulated. For example, bacteriophages will use RNA or DNA to replicate within the bacteria. Another one is Saccharomyces cerevisiae. They will use the RNA for the protein synthesis and for the gene expression. So that is how the nucleic acid will play a role in the microbial system. Especially it helps in the genetic information storage, helps in the protein synthesis, and helps in the gene expression. Gene expression means helps in the passing of genes according to the environmental conditions. Is that clear?
Yes, ma'am.
Now, targeting nucleic acids, especially in the drug development. Here also, there will be inhibition of DNA and RNA for the replication of the. That is, for the drug development, like fluoroquinones inhibit the bacterial DNA gyrase, while ramifin inhibit the RNA polymers. So specifically, they will inhibit the DNA or RNA for the production of drugs. For example, antiviral agents like drugs like Acrevir target only the viral DNA and that will be synthesized. Whereas for the gene expression and modulation, only the targeting bacteria and the fungal ribosomes will to prevent the protein synthesis. So only the targeting DNA or the RNA will be undergoing the inhibition and helps in the production of drugs. Is that clear?
Yes, ma'am.
Now we'll see some of the examples like successful drugs which has been developed. One is penicillin drug, which will target the bacterial cell wall and which will undergo the synthesis. That is how the penicillin has been produced. Another one is echinocandins, which will target the fungal cell wall, which undergoes the synthesis. Then in the rifampicin, which will inhibit the bacterial RNA polymers without affecting the human body's RNA polymers. So these are some of the successful drugs which has been developed with the help of this organic molecules. Is it clear?
Yes, ma'am.
Yeah. Now we'll move on to the microbial metabolism and the growth. So microbial metabolism refers to the biochemical process which will occur within a microbe to generate energy and to build the cellular structure. And microbial growth will refers to the increase in the number of cells which requires energy, nutrients, and proper environmental conditions. So now we'll see how the microbes will undergo different metabolisms, that is different biochemical processes, and how the microbes will grow. Okay.
Okay, ma'am.
The first one is microbial metabolism. So as I told you, it is a set of life-sustaining chemical reactions which will occur in an organism, which can include catabolism, like breakdown of different molecules for energy production, or anabolism, that is building up of different molecules for simpler ones, from the simpler ones. So there are different metabolisms which will undergo. So if you see the metabolism pathway, you can see first one will be a catabolic pathway. The catabolic pathway is a breakdown of complex molecules into a simple one in order to release the energy. So this is a one way of metabolism happening in the microbes. Understood? One is catabolic pathway.
Understood.
Breakdown of
Understood.
molecules to release the energy. Is that clear? Catabolic pathway is clear?
Yes, ma'am.
Second one is anabolic pathway. Anabolic pathway means there will be synthesis of this complex molecules by using the energy. So these are the two ways in which the metabolic pathway will be taken up. Next one, energy sources for the microbes. One energy source is chemotrophs, which will obtain the energy from the chemical compounds, and the another one is phototrophs, which will obtain the energy from the light. So these are the different ways in which the microbial metabolism will take place. Is that clear how the microbial metabolism takes place? One is catabolic and anabolic pathway. Another one is chemotrophs and phototrophs in the way they consume the energy source. Is that clear?
Yes, ma'am.
Yeah. Next one is metabolic pathways. One is glycolysis. Glycolysis is a breakdown of glucose to produce the pyruvate and ATP for the energy production. And this will occur in the cytoplasm or for both aerobic and in the presence of anaerobic organism, glycolysis will take place. So this is another metabolic pathway where there is breakdown of glucose to produce pyruvate and energy, that is ATP. Another one is citric acid cycle, or which is a commonly called as Krebs cycle, which you have learned in the biochemistry, which is like further breakdown of your pyruvate to generate energies. That will occur inside your mitochondria or inside your cytoplasm. So this is another method of metabolic pathway occurring in the microbes. This is clear. I hope you have learned this glycolysis pathway, Krebs cycle pathway in your biochemistry. You can refer your biochemistry for the same. Okay. Clear?
Clear, ma'am. Clear.
Yeah. Next one is ETC, electron transport chain. It's another way of metabolism happening. That is your ATP synthesized via oxidative phosphorylation using the electrons. And this will occur in the mitochondrial membrane or inside the plasma membrane. Last one is fermentation. Like in the absence of oxygen, some microbes will use fermentation in order to produce the energy and convert this pyruvate into lactic acid or ethanol. So these are the various steps in which the metabolism will occur in the microbes. One is fermentation, second one is electron transport chain, third one Krebs cycle, fourth one glycolysis, or the common metabolic pathway like catabolic or anabolic phase. Understood the different pathways? Understood? Different pathways, how
Yes, ma'am. Yes.
it happens?
Yes.
Now we'll see the microbial growth. So as I told you, microbial growth is the increase in the number of the microbial cells or the, microbial cells and not the size. So microbial growth is either exponential with each cell dividing into two and leading into doubling the effect. So if you see a bacterial growth, there is a growth curve you can see. And in the growth curve, if you see, there is a lag phase where there is no division of the microbes will occur. When they adapt to a new environment, there won't be any cellular division. That phase we call it as lag phase. Second one is exponential log phase. Exponential phase means where there is a rapid cell division and growth of microbes will be seen. Next one is a stationary phase. Stationary phase means the growth will be equal to the death rate. How much microbes will be grown will be equal to the how much microbes are being dead due to any nutrient depletion or due to any waste accumulation. Next one is death phase. Death phase means there is a nutrient exhaustion and toxic buildup which can lead to death of the all microbial cells. Another one is generation time. Generation time means how much time it will take for a microbe to develop or microbial population will increase in their number. So these are the things you can see in a growth curve of a microbe. Is it clear?
Yes, ma'am.
So one is a lag phase where there is no cellular division. Second one is exponential phase where there is a rapid cell division and growth. Stationary phase where there is growth rate and death rate will be equal. Death phase where there is a nutrient exhaustion and buildup of toxic materials. And generation time means the time which takes for a microbial population to multiply or increase there in the number. So this is how the microbial growth will be calculated. Clear?
Yes, ma'am.
Yeah. Now we'll see what are the factors which will influence the microbial growth. One is temperature. So microbes are classified into different types according to the temperature range. Like some bacteria will grow in a hot temperature, some bacteria will grow in cold temperature. Like that, temperature differences also matters. So you can classify the microbes into psychrophiles. Psychrophiles means those microbes which live in a cold condition. And the another one is mesophiles, where there are moderate temperature the microbes will prefer. Another one is thermophiles, where the microbes will live in a heat condition. So this is based on which the microbes will grow. Understood? The first classification is according to the temperature. One is psychrophiles, which microbes grows in a cold condition. Mesophils, where there is a moderate temperature, microbes will grow. And thermophils, where microbes will grow in a hot condition. Second thing factor which influence the microbial growth is pH level. So most microbes will grow best in a neutral pH where the pH level is 7. And but some will be adapted to the extreme pH values. So you can again classify them into acidophiles and alkalophiles. So acidophiles where the environment will be acidic in nature, and the alkalophiles where the environment will be alkaline in nature. So this is the second factor which will influence the microbial growth. Is it clear? Temperature and pH is clear?
Yes, ma'am.
Yeah. Next one is osmotic pressure. The concentration of solute will affect the water movement across the cell membrane. Like always, there is a movement of water molecules from a high region to the low region. And then you can classify the microbes as halophiles, like the microbes which can tolerate and require high salt concentration, which will live only in the salt water. That is called as halophiles microbes. Next is the oxygen availability. Oxygen availability means some requires oxygen for their growth. They are called as aerobe microbes. Some will grow in the absence of oxygen. They are called as anaerobes. Some will facultative anaerobes. That is, they can grow with or without oxygen. And some require low level of oxygen. They are called as microaerophiles. So based on the availability of oxygen also, growth of microorganisms will affect. Is this clear?
Yes, ma'am.
Now, another factor which will affect the growth is the nutrient availability. So, microbes also require specific nutrients for its growth. So there are classified into macronutrients and micronutrients. So macronutrients will include carbon, nitrogen, sulfur, phosphorus, etc. And micronutrients will include iron, zinc, magnesium, etc. So nutrient availability is also required for the growth of the microbes. And growth medias, like some will grow in a solid culture, some will grow in liquid, some will grow in a semi-solid environments. So that will provide nutrients for the microbes to grow. So this is what nutrient availability which is required for the microbial growth. Next one is moisture content. Water is essential for the microbial cells to function, and that includes metabolism and enzyme activity. So moisturizer is also a factor for the growth of the microbes. Last one is light. Light is an important for photosynthetic microbes, and some microbes can be inhibited or damaged by the UV radiation. So some require light and some doesn't require the light. So these are the some of the factors which will influence the growth of microbes. Is that clear? Factors affecting the growth of microbes. Is it clear?
Yes, mom, it's clear.
Next one, application in a infection control. Like what are things you should take precaution when you are coming across any infections. So one thing is when you are learning about the microbes and when you are doing anything in the laboratories, first thing you have to maintain is environmental hygiene. Like regular cleaning, disinfections will minimize the microbial reservoirs. So wherever you touch, you have to clean the surfaces so that environmental hygiene can control the infection. Second thing is sterilization and asepsis. Asepsis means sterile techniques. So sterilization and asepsis is also important. Like ensure all the instruments and the environments are free of microbes. So if you are testing any microbes or if you are growing any microbes, all the instruments which you use has to be sterilized so that you can control the infection. Third one is proper hand hygiene. So prevents the direct transmission of pathogens. Like unknowingly you touch the microbe and you touch your nose, mouth or something, it can enter the human body. So proper hand hygiene is also a factor to control the infection. Next one is proper storage. Like food, medications and medical tools must be stored in such a condition where unstable for the microbes to grow. So if you see, there are fungal infection growing around the medications or in the foods and all, right? So that also you have to take care. So if the fungal infection you see in any food items, you have to throw them out so that you can control the infections. Last one is monitoring the conditions, like maintain a controlled temperature, humidity and pH in a healthcare settings to limit the microbial proliferation. So if there is too much of moisture content inside a room where there is no ventilation, the microbes can grow. So you should clean each and everywhere in order to limit microbial growth. So in such a way, you can control the infections. Understood? Infection control. Is it clear? The infection
Yes,
control
mom.
is clear?
Yes, yes.
Yeah. Now we'll see the various pathogenesis and host interactions, like different stages of infectious diseases and immune escape, evasion. Okay. So usually if you see there are different stages for the infectious diseases. One is during incubation period. When you incubate the organisms, like pathogens will, when the pathogens enter the body and they try to begin to multiply in your body. This is the first stage for the infectious disease, right? That is called as incubation period. That is the period where the pathogen will enter the body and try to multiply. So in such stage, what the nursing has to do? Nursing has to focus on monitoring of the early signals of infection and educate the patients on the prevention methods. Understood? So the very first one is incubation period. So in that period, the nursing has to focus on
Yes, mom.
early signs and infection and you have to educate your patient. Second one is prodromal stage. That is one set of a non-specific symptoms. Like unknowingly, there will be symptoms like fever, fatigue can be seen. So there, what the nurses has to focus is you have to early detect what are the signs and symptoms, and you have to do the management. Third one is illness stage. Like there is specific symptoms will be manifested. Like pathogens will load peaks. That is, it will multiply in your body like anything, and there will be a lot of symptoms can be seen. So in such condition, the nurses has to focus on what are the supportive care, treatment, and what are the prevention methods to prevent the complications has to be focused. Understood? Fourth one is
Yes, ma'am.
convulsion stage.
Understood.
So convulsion stage means the recovery and restoration of the health. That is, nurses has to focus on the rehabilitation, patient education on preventing the recurrence. The patient should not get affected again. So you have to educate the patient in such a way that they won't get affected anymore with the same disease. Is that clear?
Yes, ma'am, it's clear.
Yeah. Next is pathogen immune evasion mechanism. Like how can you prevent or escape this immune pathogen mechanism? So one is antigenic variations. One thing you can do is change the surface proteins to escape the immune detection. So if you change the surface proteins, then they will not be able to detect the pathogen. Second thing, inhibiting the phagocytosis. That is capsule production or toxin production, which can inhibit the phagocytosis production. Third one, hiding the inside host cell. Like intracellular pathogens like viruses can be hidden. Fourth one, suppressing the host immunity, like producing proteins that will interfere with the immune response. For example, HIV, mycobacterial tuberculosis, etc. in such a way that you can reduce the pathogen immune evasion method. Like pathogen will not be that much effective. Okay, understood?
Okay. Yeah.
Yeah, just read it once and see.
Sorry.
Okay. Now we move on to the parasitology. So till now we were learning about the microbiology, different life cycles, different carbohydrates, proteins, lipids, which are the different factors affecting the metabolism of microbes, life cycle, growth, etc. Now we'll move on to the parasitology. Parasitology will involve all the different types of life cycle, how that interaction occurs, etc. Fine. Now we move on to parasitology. We'll start
Yes,
parasitology.
ma'am.
Yeah. So what is parasitology? So it is a scientific study of different sites of parasites, their biology, their life cycles, and interaction with the host body. So usually parasitology will contain the life cycles, which will refer to the various sequence of the developmental stages happening in the parasite, which will undergo complete its life. Often they involve one or more hosts in the life cycle. And that stages will include laying of eggs, formation of larvae, then that is developing into the adult form, and then that will reproduce. So this is how the life cycle will takes place. And then host interaction, which will describe the relation between the parasite and its host, which can induce a host providing nutrient, shelter, or an environment that parasite reproduction will take place and the parasite growth also will take place. So usually if you take a life cycle of anything, host will be a human body. So inside the human body, they will get the nutrients, they will get a shelter, and they get an environment where they can multiply and they can grow. Understood? This is what we discuss in the
Yeah.
parasitology.
Yes.
Is that clear?
Yes.
Yeah. Now we'll see the parasite life cycle and the host interaction. As we have discussed the same thing before, malaria parasite. When the disease is caused is malaria by the species Plasmodium species. So how was the life cycle in the same is one human will be the host. right? So what will happen is the sporocytes will be injected into the human body once the bite is occurred by an infected Anopheles mosquito, right? So when the Anopheles mosquito who is infected, when they bite the human body, they will inject the sporocytes into the human body. And this sporocytes will migrate into the human liver, and there they will develop into merocytes. And then that will enter the blood cells, that is in the RBC. And this merocytes will try to multiply inside the RBC, and then they will cause the rupture, and that will be released more merocytes, which will lead to the fever cycle. Like they will try to reproduce and they will multiply inside the human body. So that is what happens in the malaria parasite. And here another one will be the next size will be the mosquito. When an unaffected mosquito will come and bite this human, they will get this infection. So the mosquito will ingest this gametocytes during a blood meal, and there the fertilization will occur in the mosquito's gut. And again there is production of sporocytes, and that will migrate into their salivary glands. Again that will bite on another human body. Again this life cycle will continue. Understood the parasite of malaria parasite life cycle? Understood?
Yes, mom.
Just read it once and see if you understood the same. Like how the malaria will be caused, how that life cycle is taking place. So first one, unaffected human will be a host. When an affected Anopheles mosquito will bite the human body, they will inject all the sporophytes into the body. That will migrate into the liver, and then they will develop into merocytes. Then that will enter your RBC. Inside the RBC, this will multiply and that will rupture and release into the body, which can cause fever cycles. Then what will happen is they will try to produce eggs also. Then another mosquito, unaffected mosquito will come and bite. So they will ingest this gametocytes from the human body during the blood meal. And then the fertilization occurs inside the mosquito's gut. And then again sporocytes will be produced and that will be migrated in their salivary gland. And then again this has been continued. Understood? Understood the parasite life cycle?
Yes, mom.
So here, what are the prevention tips you can give being a nurse? You can educate your patient on using the insect repellents or bed nets or any strategies to eliminate the mosquitoes breeding. And you can emphasize the importance of completing the anti-malarial medication regimes also. So this is what the prevention tip being a nurse you can give to your patient. Clear?
Yes, mom. Yes.
Yeah. Next is Helminthes life cycle. As we discussed before also, first there will be a direct cycle like some Helminthes like Ascaris lumbricoides will have a simple life cycle which involves only one host. So what happens here is eggs will be ingested through the contaminated food or water, and then that will hatch into the larva in the intestine, and then that will grow into the adult, and that will release eggs into the environment. Then the tapeworms, which will involve an intermediate host, there the larva will be ingested by this tapeworm, and then this undercooked meat, when it is eaten by a human body, then that will grow inside the human body's intestine. So here what prevention method being a nurse you can give is promote the hand hygiene and proper food handling methods or practices you can educate your patient. And you can advise them to have a periodic dewormings in the endemic areas. You can ask them to take up the medications. You can ask them to promote proper hand hygiene and proper food handling. So these are the tips you have to give for your patients. Understood?
Yes, mom. Yes.
Yeah. Next one is protozoa. In their life cycle, what is happening is again indigested cyst will enter the body, which is either it will be in the contaminated food or water, it will enter the body. And then they release the trophocytes into the small intestine and then causes diarrhea and malabsorption in the human body. So in such condition, being a nurse, you can advise the patient to have a safe drinking water practices, proper sanitation, and you can ask them to be a stress the need for water, fruits, vegetables thoroughly. Like ask them to wash everything before they consume anything and everything. Ask them to wash it properly and ask them to have a safe drinking water practice. Understood? These are the prevention tips being a nurse you can give to the patient. Is that clear? Is that clear? Clear?
Yes. Yeah.
Yeah. Next, we'll see what are the role of nurses in this parasite management. How and how you can manage your patient. One thing you can help them or you can advise them to have anti-parasitic drugs. So administration of anti-parasitic drugs, you can ask the patient to take up the anti-parasitic drugs. So administrating appropriate anti-parasitic drugs based on the therapies for malaria or helminthes, whatever, you can monitor for the side effects and the patient adherence. So in such a way that you can help your patients. Second thing is health education. You can teach different communities or people or the about the personal and the requirement of environmental hygiene. And you can also encourage the people to have a routine health checkups in the endemic regions. Third thing is preventive care and surveillance. Like you can collaborate in different vaccination programs and you can advise your patients to take up a regular vaccinations. And you can also support efforts to track and eliminate the local outbreaks. You can understand, you can identify which organism is causing the infection and you can support that people also. So these are the some of the role of nurses in the management. Understood? One is you can ask, you can administrate the people with the anti-parasitic drugs. Second thing, you can give them education. Third thing, you can collaborate in vaccination programs and you can support the and eliminate the outbreaks of different microbes. Is that clear?
Yes, mom. Yes.
Okay. Next is different diagnostic techniques and laboratory safeties. Now, how and all you can diagnose the and how you can do the laboratory techniques also. So as I told you, in order to study the microbe, they do something called as stainings. So staining is a method which is essential in a microbiology for enhancing the contrast of a microorganism, making them visible under a microscope. So if you want to learn about any microorganism, you have to keep it under the microscope and it should be visible. So for that visibility, you will make them stain. So there are different techniques of stainings are there. One is simple staining and another one is differential staining. So if you see what a simple staining is, mainly the purpose of simple staining is to highlight the entire microorganisms so that you can study what is the basic morphology and how that microorganism is being arranged, everything. So in order to highlight that organism, you do a simple staining. Second thing is differential staining. Here, the differential staining is for differentiating between the different types of bacteria or different cell structures. There are different types of bacteria, eukaryotic bacteria, prokaryotic bacteria, unicellular, multicellular. So in order to differentiate the types of bacteria, we can go for differential staining. And under differential staining, there is a gram staining, which is gram positive and a gram negative staining. So what is gram staining is the principle is to differentiate the bacteria based on the cell wall structure. So for that you use the gram staining in order to differentiate the bacteria based on the cell wall structure. You do gram staining. So in the gram positive staining, you can see a thick peptidoglycan layer will retain crystal violet stain. So that will appear purple in color, whereas in gram negative, there is a thin peptide glycan layer will lose the crystal violet, but maintain a saffron color. It will appear a pink color. So as you see in this image, when there is a thick layer being coated, there is a purple color staining you can see. Where there is a thin layer of peptide glycan has been coated, then you can see a pink color staining. So this is a gram positive staining and this is a gram negative staining. Understood gram staining? Understood? So the one way of diagnosing or learning the microbe is by the process of staining. Staining means just to make them visible under the microscope. This can be either a simple staining or a differential staining. Simple staining is just to highlight the microorganism. Differential staining is to differentiate what type of bacteria or what type of cell structure do they have. And in that you have gram staining method, that is positive gram staining and negative gram staining. So if it is a positive gram staining, you can see a purple color appearance. And if it is a negative staining, you can see a pink color staining. Is it clear? Gram staining? Gram staining?
Yes, ma'am. Yes.
Okay. Next one is culture techniques. Like you need to grow the microorganism, right? So you give culture, such as growing microorganisms in a controlled environment in order to identify and in order to study what pathogen it is. So in the laboratories, if you see, they will try to grow these microorganisms, isn't it? So that requires different media for the microorganism to grow. So culture technique is a growing of microorganisms in a controlled environment to identify and to study the pathogens. So if you see, there are different media are provided in order to grow the microorganisms. The one media is selective media. Selective media means it will support specific organisms which by inhibiting the others. Another media they provide is differential media, which will identify the organism based on the biochemical properties. Another one is enriched media, which will provide nutrients for the fastidious organisms. And this is usually applicant in the blood culture, throat swabs, and in the urine culture. We do this technique. Understood? Culture techniques we do in order to grow the microorganisms in the labs. So they require media to grow. So one media is a selective media where they will support the specific organisms by inhibiting the other. Only the specified organism will grow and the rest of the microbes will die off. Second one is differential media that will identify the organism based on their property, biochemical property. They can identify the growth. Third one is enriched media. That is, the media will provide all type of nutrients for the fast growth of the organism. And this is applicable in the blood culture, urine culture, as well as in the culture of throat swabs. Is it clear?
Yes, ma'am.
If you see in the image, in this plate, they will grow the organisms by putting the culture. By providing the culture in this plate, they grow the microorganisms. Understood?
Yeah.
Next one is molecular diagnostic techniques like polymerase chain reaction, PCR, which will amplify the DNA for the pathogen detection. That is during the COVID outbreak, if you see, they were taking the throat swabs and all, right? So that you can do PCR test. So they will amplify the DNA for any sort of pathogen detection. Another technique of diagnostic method is real-time PCR or RT-PCR, which will detect the RNA virus like SARS virus or COVID virus, etc. So this is advantage of doing this molecular diagnostic technique is they are highly sensitive and specificity and the speed of diagnostic method, like easily they will give the results. So molecular diagnostic methods will include polymerase chain reaction, that is PCR reaction, which will amplify the DNA for the pathogen detection. Another one is real-time PCR. Real-time PCR is a RT-PCR test which will detect the RNA virus like SARS or COVID virus. Last one is serology test. Serology test will include ELISA test, immunofluorescence test, or any agglutination test. What is ELISA test? What is ELISA test?
I don't know. Is it supposed to be somebody's name? Immunofluorescence.
Enzyme-linked immunosorbent assay, right?
Yeah.
Yeah. So for that and all serology tests you can do is either ELISA test or immunofluorescence test or any agglutination test. So this is applicable especially in the detection of any antibodies or any antigen when it is entered your body. For example, HIV testing, dengue testing, for that and all you can go for serology test. Understood? Different diagnostic
Yeah.
techniques. Now we'll see what are the different laboratory safety mechanisms required. So as I told you, when you're growing any microbes in the lab, you should take precautions, right?
Sure.
Yeah. So we'll see what are the precautions to be taken. One thing, you have to ensure there is a safety in the diagnostic labs is critical to prevent the contamination, infection, or any sort of accidents. So you have to prevent any contamination or infections or any accidents occurring in the labs when you're growing any organisms. So very first thing you require in a lab for the your safety is personal protective equipment, PPE kit. As you see in the image, you can see them wearing a coat, a glass, then gloves, shoes, and everything. So this all these things together we call it as a personal protective equipments. So this is essential. Which essential to wear this PPE kit, which includes a lab coat, gloves, mask, goggles, face shield. So when you are growing a microorganism, unknowingly or accidentally, if it falls on your body, it should not affect your body health. So one safety mechanism is wearing a PPE kit. Understood?
Yes,
Understood?
ma'am.
Yeah. Second thing is
Yes, understood.
the specimens. You should take utmost care when you are handling the specimens. That is, you should always follow aseptic techniques, like sterile techniques has to be taken to prevent the contamination of any microbes, like proper labeling, proper transportation, and proper maintenance of the specimen integrity has to be taken care when you're handling the specimens. Next thing is laboratory waste disposable. Like obviously you have to dispose the waste. So that also can cause contamination and growth of microbes. So always the biohazard waste, whenever you are disposing, you have to do it autoclave. Autoclave means sterilization you will do or incineration of the cultures, tissues and infectious materials. Whenever you dispose the waste, you have to classify them into dry waste, wet waste, and you have to separate them for autoclaving also. For example, sharp objects, if you are discarding, that has to be discarded in such a way that it doesn't cause any problem to the environment. For example, disposal of any puncture resident containers, like in separate containers, you have to throw the sharp materials like any needles or any blade or anything. If you're disposing, that has to be disposed in a, such a container which doesn't cause any puncture. Then when you are disposing any chemical waste, you have to neutralize it before the disposal. Like that, it should not cause any contamination to the environment also. So you have to neutralize it before the disposal. So that is how you have to dispose the waste. Is that clear? Waste disposable?
Yes, ma'am. Yes.
Yeah. Next is emergency protocols to be followed, like spill response, like use of any absorbent materials and disinfectants. If suppose by mistake any bacterial microbe has been fallen from your hand, then that area has to be cleaned immediately with the disinfectants. You have to clean the surface. Then reporting and managing the accidental exposure. always you have to immediately report whatever accidents has been occurred from your side. Second, last thing is equipment maintenance, like regular calibration of the diagnostic equipments, cleaning and sterilization of your reusable tools also has to be taken care for the safety. Understood?
Yes, ma'am.
So as I told you, waste segregation, you have to segregate the waste before the disposable. Like you have different containers with different color codings so that you can dispose the waste in different containers. Usually you have a black color container in the hospitals to throw all the plastic waste. Then you have a red colored container, you can throw all the contaminated or infected waste materials. Then you have a yellow color also again for a disposal of any infected materials. Then you have a blue color where you can all the syringes, gloves and everything. Then you have a white color container where you can throw all the sharp materials like needle, blades, everything. Understood? So these kind of waste segregation is very important in the hospital sites before the disposal of the waste. Clear?
Yes, ma'am, clear.
Next is infection control and sterilization. Like how you can control the various infections and how you can sterilize the equipments. So infection control is the prevention and the spread of infections in the healthcare settings. Like you will prevent the spread of infections in the healthcare system is the infection control. Sterilization is the complete elimination of all the microorganisms, including the spores. Third one is disinfection, that is reduction of any sort of harmful microorganisms in the surface of the objects. And then you have aseptic techniques, that is practices like sterile practices to minimize the contamination during the medical procedures. So these are to be followed, always to be followed in a hospital setup. One thing, you try to control the infection. Secondly, you should always sterilize your equipments, whatever you use. Third thing, you have to use disinfectant to clean the surfaces. And last one is always follow sterile techniques for controlling
Disinfect.
understood. Being a nurse, you follow these guidelines. Understood?
Yes, ma'am.
Now we'll see what are the different sterilization techniques available. So sterilization is nothing but the process of destoring all the microbial life, including the spores. So that is what we call it as a sterilization. Process of destoring, destroying all the microbial life, including the spores. So you will destroy or you will kill all the microbes when you are sterilizing the equipment. So method of sterilizing, if you see, one is autoclave. As you see in the image, this is an autoclave machine. So inside this machine, you put all your equipments and you keep it under a steam. So under that steam, that tools will get sterilized. So that is what we call it as autoclaving. Under steam, that pressure, the equipments will get sterilized. Second thing is dry heat sterilization. Is another method of sterilizing the equipments. Another one is radiations. You can pass on the UV radiation on the tools and equipments to sterilize the same. So these are some of the physical methods of sterilizing. Understood the physical method of sterilization? Autoclave, dry heat sterilization, and radiations. Then there are chemical sterilizations are also available, where you use chemicals like ethylene oxide gas, ETO, or hydrogen peroxide vapors. So these are the another ways of sterilizing the equipment. So application, where does this kind of sterilization you can see is, one is surgical instruments. When they are sending it for sterilization, they do autoclave or they do this chemical sterilization. Or laboratory tools or any medical devices, when you want to clean them, you do this physical or chemical sterilization methods will be followed. Understood? So usually in the exam, this autoclave or the sterilization method has been asked. Okay? Is it clear? Sterilization technique is clear?
Yes, mom, it's clear.
Yeah. Next one we are discussing is about the disinfectants. Okay. Do you have any idea about the disinfectants?
Sorry, mom.
Disinfectants. What are disinfectants?
They are reducing harmful microorganisms on surfaces.
Okay, so disinfectants are the ones which will reduce the harmful microorganisms on the surface. So in the sterilization, we learned that they will kill the microorganism. So whereas in disinfection, you don't kill, you just reduce the harmful microorganisms on the surface. So there are different types of disinfectants are available, like high level disinfectants, which can kill most of the pathogens, except some spores. Like 99% of the microbes will be killed when we use high level disinfectants. Second thing is intermediate level disinfectants, which will kill the bacteria and fungi and the viruses. For example, phenolics, alcohols, and etc. Last one is low level disinfection. So low level disinfection will kill some bacteria and the viruses. So where does this disinfectant application comes is especially in the patient care equipments, environmental surfaces. For example, patient bed you clean or the handrails of the door or any patient equipments, etc. If you want to clean, you can use disinfectants. Understood?
Understood.
Yeah. So these are the some of the sterilization and disinfectants you can see in the hospital. Next is aseptic technique. So aseptic technique is nothing but the sterile technique. Sterile technique, that is practices you follow in order to prevent the contamination by the pathogen. Like you will try to minimize the contamination with the pathogens. So that is what aseptic technique is, sterile techniques. So what are the key techniques is, one thing is proper hand hygiene, like hand washing and use of any hand sanitizers. That is why in the hospitals you can see every corner there is a hand sanitizers are kept. So every time when you touch the patient or even if you touch the handrails or even if you touch the patient bedside also, you have to use the sanitizers. So that's a one way of aseptic technique. So hand hygiene is a very important technique. Second thing is use of a PPE kit. PPE kit which contain the gloves, mask, gown, shoe cover, head mask, googles, face shield, etc. for your protection. Personal protective kit. Next is sterile field setups during the invasive procedure. Like you should not come to the sterile field where only the sterile things are being kept so that it will prevent the contamination. Next one is proper handling and disposal of any sharp objects. As I told you, you have to segregate and then you have to dispose the same. Now, where does this aseptic technique application comes is under patient wound care, patient catheterization, when the catheter is inserted into the patient body. If you don't provide aseptic technique, then there will be infection occurring to the patient. And in the surgical procedure, you provide a sterile field where for application of sterile techniques. Is that clear? Is that clear?
Yes, mom, it's clear.
Next is what is the nurse role? What will be your role in the infection control in the hospitals or anywhere? So one is critical responsibility includes ensuring adherence to the sterilization and disinfection protocols. You have to stick on to the various sterilization and disinfection protocols in order to control the infection. Second thing you have to do is you have to educate your patients and other staffs in the hospital regarding the hand hygiene, importance of hand hygiene, and importance of infection control practices. Third thing, you have to monitor the compliance with this aseptic techniques. You have to follow the aseptic technique practices in the day-to-day life. The next one is identifying and isolating infectious patients when necessary. When you confront that a patient is infected with any viral infections, you have to isolate them so that you can prevent the infection. Next is reporting and documenting the infection outbreaks. So you have to report to the higher authority and you have to document which kind of infection was outbreak and what precautions have to be taken, etc. So these are some of the major role of nurses in order to control the infection. For example, setting up a sterile field, supervising the equipment sterilization, and promoting vaccination importance. In such a way also you can control the infections. Is that clear? What are the roles of nurses in the control of infection? Understood?
Yes, ma'am. Yes.
Read once and see.
Sorry.
Okay, next we'll see some of the practical applications of infection control in a nursing. Okay. So practical
Okay.
yeah. So practical application, if you see, it is practical application of an infection control in a nursing is nothing but like how which or what are the practices you will follow in order to control the infections. So one thing is infection control practices are curative for preventing any healthcare associated infections like hospital-borne infections. So infection control practices are very important to be preventing further healthcare associated infections and ensuring the patient safety. The very first thing you have to do is in the wound care. When you are giving a wound care to any patient, what are things you should follow? That is wound care. You have to perform hand hygiene before you do any dressing or before you touch the wound. Being a nurse, you should perform a proper hand hygiene before as well as after the wound dressing. Next thing you should do is use sterile equipments and aseptic techniques for the treating the wound. Third thing, you have to properly clean and dress the wound so that it doesn't cause any other infection to any other body. So what is the importance here is to minimize the risk of wound infection and to promote a faster healing of your patient. An example here you can give is preventing surgical site infections in the post-operative care. If there is any surgical wound that has to be taken care, you have to follow all these things. That is, you have to perform hand hygiene before and after touching the patient, use sterile equipments for dressing, and use aseptic techniques like sterile techniques to take care of the wound. And you have to clean the dressing in such a way that it doesn't cause any infection or any problems. Is that clear? Is that clear? Is that clear? Are you there in the class?
Yeah, my network just went off, but it's back now.
Is it clear?
Yes, it's clear.
Okay. Next one, catheter care. When you're putting a catheter into a patient body, as you see in the image, when you are putting a catheter or external thing into the human body, again you have to use a sterile technique during the catheter insertion. As you see in this image that the patient is being put in under the catheterization by a nurse, and the nurse has wore gloves when he or she is touching the catheter. So you have to use sterile techniques during the catheter insertion. And there should be a regular cleaning of the catheter site with the antiseptic solution. So wherever the catheter is inserted in the body, that body region has to be cleaned with the help of antiseptic solutions like Dettol, Savalon, everything. Then you have to avoid the prolonged catheter use to reduce the risk of infection. So if you have put the patient on a catheter, it has to be removed after some time. Otherwise, it can cause infections. These are the nursing care you should give when you're putting a patient on catheter. Now, what is the importance is to prevent catheter-associated urinary tract infections. An example for Foley's catheter, that is urine catheter. When you put a urine catheter, you have to take care in order to prevent any sort of biofilm formation. Like it should not cause any infection in the patient body. So this also has to be taken care when you are putting any patient on a catheter. Understood catheter care?
Yeah, yes, ma'am, yes.
Next thing you have to follow is hand hygiene. As I told you, the WHO, that is the World Health Organization, has given 5 moments of hand hygiene. Like you have to hand wash before you touch a patient. You have to hand hygiene has to be maintained before you do any aseptic procedures. After the body has been exposed, the body fluids has been exposed. If it touch by mistakenly, also if it touch any body fluids, then also has to hand wash. And after touching the patient surroundings like patient bed, patient side, anything if you touch, again you have to do proper hand washing. So use of any alcohol based hand rubs or washing hands with a soap and water is recommended to have a proper hand hygiene. Now what is the importance of the same is to reduce the transmission of any sort of pathogens between the patient as well as between the healthcare workers. That is why even if you go to the patient side also, you have to sanitize your hands or directly have to wash it with the soaps. Understood hand hygiene?
Yes, ma'am.
These are the different steps to hand hygiene. So you have to wash your hands, then you have to put the hand rub, you have to rub it your palms on the upside, downside, your fingers, your thumb, your palm. Then you have to wash it and then you have to dry it. So this is how you perform the hand wash practice. Understood the hand wash practice? Just go through the hand wash practice once. Just go through it.
Yes.
Because read it once, hand washing practice. Just read it. Done. Here.
Yes, clear, ma'am, very clear.
Okay. Next we'll see a case study. Okay. So here the case study is regarding a MRSA. What do you mean by MRSA? Methicillin resistant Staphylococcus aureus. Like a person become resistant to the methicillin due to Staphylococcus aureus. So here the background is an hospital acquired infection, HAI, hospital acquired infection caused by an antibiotic resistant bacteria, common in the surgical wounds and catheter sites. So here that is a background or that is a case. Okay, the patient has got a hospital acquired infection due to the antibiotic resistance. So the patient cannot be take any sort of antibiotics because he or she is resistant to that bacteria. And this kind is very common in case of a surgical wound or in the place of catheter sites. So here the challenge is there is a very limited treatment option available due to the antibiotic resistance. So what prevention methods being a nurse you can give us? First one, you have to adherence to the hand hygiene protocols.
Yes, ma'am, yes.
Yeah, you have to teach the patient to have a proper hand hygiene protocol. Second thing, you have to advise the patient to have a proper cleaning and decontamination of the surface. Wherever the surface the patient has been, or if you are going and touching, wherever that is, you have to clean that surface with the help of disinfectants. Then you have to isolate that infected patient separately so that further contamination or infection will be prevented. Understood? So in such condition, these are the prevention methods being a nurse you should take. Okay. Just read a case study. Just read this case study once. Just read this case study once. Done? Understood?
Yes, ma'am, understood.
Okay. Now we'll move on to another case study. Here, the infection is due to the species that is Floristeron difficult. So here the background or the case here is there is spore forming bacteria is causing severe diarrhea and colitis in the patients. Okay, because of the spores, this bacterium is causing severe diarrhea and colitis. And this is associated with the prolonged use of antibiotic. So here the challenge is spores are very resistant to the disinfectant. Even if you use disinfectant also, spores will not die. Okay, in such a way, what prevention you can do is, you can advise your patient to use soap and water for hand washing. So if you hand wash for at least for a 5 or 10 seconds also, it can kill all the spores. So you can usually use alcohol-based rubs or any infectives for the proper hand wash. Second thing, you can implement a contact precautions. Like any sort of handshaking can be prevented so that there won't be any passage of infections. Third one is you can ask them to have a proper environmental cleaning with the sporocidal agents. So you can ask them to clean their environmental surfaces with any sporocidal agents which can kill the spores. So these are the techniques or the prevention methods being a nurse you can advise. Understood? So here the disease is caused by the spore forming bacteria, which is causing severe diarrhea and cholestasis in the patient. And here it is mainly caused. So here what prevention method being a nurse you can give is ask the patient to use hand wash, like with a soap and water, you have to ask the patient to hand wash. Second thing, usually the alcohol-based hand rubs are effective. Now, implement the contact precautions like handshaking can be avoided. Then proper environmental cleaning with the sporocidal agents which can kill the sporocytes. So these are the prevention methods you can give for your patient. Understood?
Yes, ma'am, understood.
Yeah. Can you just read this once? Read and see this case study.
The background is noisy.
Sorry.
Okay. The background is very noisy. Let me leave noise.
Okay. Can you read the slide once? I mean, just read and see if there is any doubt. Just read and see if there is any doubt.
Okay, can I read now? Please display on the screen.
Sorry.
Okay, you say I should read case study number 2.
Yeah. Is it clear?
Yeah, it's clear, it's clear.
Okay. So all the infection control practices like wound care, catheter care, and hand hygiene care are very essential in your day-to-day nursing practices. Okay. So that is what we have learned so far. So case studies like MRSA will illustrate the consequence of lapses in their practices. So call of action being a nurse you can do is nurses must consistently implement these measures in order to maintain a safe clinical environment. So you have to advocate or to teach your patients the various techniques to control this infections, and prevention methods has to be taken. Understood? Understood?
Yes, ma'am. Understood.
Class is done. So usually this MRSC case study has been asked for the exam. Exam point of view, MRSC class. So please learn this case studies like you can. Understood? Clear?
Yeah, clear.
So what are the different role in infection control? Can you tell me? What are the different nurses' role in infection control? Can you tell me? What are the different role of nurses in infection control? Can you answer? What will be the role of nurse in infection control? Can you answer? So one thing is ensuring proper sterilization and use of disinfection protocols. Okay. Second thing is educating the patients and the other healthcare staffs regarding the importance of hand hygiene and infection control practices.
Yeah.
Third, to monitor
Okay.
and aseptic. Right? Third thing, you have to isolate the whenever it is necessary, and you have to report and you have to document all sort of breaks and infections. Okay. So these are the
Okay.
nurses' role. Is it clear?
Yeah, it's clear.
Role in infection. What are the nurses' role in infection control? Can you tell me? What are the role of nurses' role in infection control? Can you answer?
Yes. Ensuring
And
adherence to sterilization and disinfection protocols. Educating patients and staff on hand hygiene and infection control.
yeah.
Monitoring compliance with aseptic techniques. Identifying and isolating infectious patients when necessary. Reporting and documenting infection outbreaks.
Very good. So the role of nurse control. Now, what are the different infection control and sterilization techniques you can follow? One is autoclave, dry heat sterilization, or a use of radiations, or use of ethylene oxide or hydrogen peroxide vapors for the sterilization of equipments. Understood?
Understood.
Now, what are the different disinfectants you can use? Either high-level disinfectants or intermediate level disinfectants or low-level disinfectants can be used to control the infections. You can follow the aseptic techniques like hand hygiene, use of PP kit, personal protective equipments, sterile field techniques, or proper disposal of the sharp objects to control the infection. Okay. Now, how the waste is segregated? Can you tell between segregation? How the waste segregation is done? How is the waste segregation done?
It's done through different waste disposal bins. You have the black, which is the black plastic container for chemical treatments. You have non-infected waste.
Done as infected and the non-infected. So usually you can
Yeah,
classify them
non-infected.
color coatings. So you can use a black color dustbin for a non-infective materials like plastics, anything. For any infective materials, you can use the red and the yellow. And any sharp objects you can put in a white color, and all the syringes, gloves, any other plastic waste you can put in a blue container. Understood?
Okay, understood.
Yeah. Now,
Network is really a problem.
parasite management is you can administer the patient with antiparasitic drugs. You can give them health education, and you can prevent care and surveillance. Everything can be given to the patients. Okay. So this is what we have
Okay,
learned in the
mom.
parasite. And under microbiology, we have learned the different stages of infectious diseases. Can you tell me which are the different stages of infectious diseases? Various stages of infectious diseases.
Incubation period. We have
Yeah.
incubation period,
Yes.
nurses focus, prodromal
Okay.
stage, nurses, illness stage, covalent stage. So
Yeah.
we
So
have
these are
incubation period, prodromal stage, illness stage, and covalent stage.
convalescent stage are the different stages. Good. Now, how you can control the infections? What are the practices for controlling the infection?
We can control infections through environmental hygiene,
Yes.
sterilization and asepsis, hand
Yes.
hygiene, proper storage, monitoring conditions.
Yes. Very good. Now, what are the factors affecting the microbial growth?
We have temperature
Yes.
and then the pH.
Yes.
We have osmotic pressure and oxygen availability.
Then nutrient availability, moisture, and light.
We have nutrient availability, moisture, and light.
Yeah. Very good. Now, how and all you can classify the microbial growth in a growth curve?
We have lag phase, exponential phase, stationary phase, death phase, and generation time.
Yeah. So lag phase is the place where you can see no cell division. Exponential phase, there is a rapid cell division. Stationary phase, the cell growth and cell death will be equal. In the death phase, there is a nutrient exhaustion and a toxic buildup can be seen. And generation time is the time taken for the microbial growth. Okay.
Okay, ma'am.
Fine. So these were the various things we have learned today. So I hope today's class has been clear. Right? Any doubt is there in the today's class? Any doubt? Today's class? Any doubt in the today's
My network is fluctuating.
Sorry?
I said my network is fluctuating.
Okay.
That's why
So
at times I can get you, at times I can't get you.
I'll send PPT and you can go through the PPT. Okay. Fine.
Okay, ma'am. Okay.
Thank you for your cooperation. Thank
You're welcome. Thank you too, ma'am. Thank you for your patience.